A very good morning to all of you, dear students, all participants, my colleagues and dignitaries. It is my great pleasure to welcome you all for this inauguration of 4th National Conference on Cellular and Molecular Biology, Recent Advances in Biotechnology. First of all, I would like to invite dignitaries on dais. I request Honorable Vice Chancellor Sir, Professor Sibaram Khara, to please come on the dais. I request Professor Parmanand Sir, Dean Academics, Dean School of Engineering and Technology on the dais. I also invite Professor B.C. Tripathi Sir and Professor Abhimanyu Jha Sir on the dais. Let's start the event with lecture of our chief guest, Professor Subra Chakravarti, a very renowned scientist who has very kindly consented to be chief guest for our ceremony. Professor Subra Chakravarti received her PhD from Jawaharlal Nehru University, New Delhi, India, and has been a visiting scientist at Yale University, USA. After a brief career as research scientist at the Center of Plant Molecular Biology, JNU, she joined National Institute of Plant Genome Research, NIPGR, in 1998 as a founder faculty and currently is serving as a director of the institute. Ma'am has several prestigious awards to her credit. The list includes prestigious J.C. Bose National Fellowship Award, Tata Innovation Fellowship Award, Award in Biological Sciences, Professor Hiralal Chakrabarti Award of ISCA, among others. She is also an elected fellow of National Science Academies in India. Ma'am is also actively associated with national and international proteomic societies. Currently, she is serving as President, Proteomic Society India. She is also serving as member of research bodies and national committees concerning education, academic activities, research funding, and policy planning in India. With these words, I welcome Professor Subra Chakravarti for her lecture.
Thank you for a nice introduction. So at the outset, I would like to thank the organizer, particularly the Honorable Vice Chancellor, as well as the Dean Academics, and in particular mention my teacher and very beloved teacher, and Professor B.C. Tripathi and Dr. Abhimanyu Jha to give me an opportunity to come and give an online lecture to Sharda University. So this is the first time I'm actually giving any lecture to Sharda, and also have never had an opportunity before this to visit the university. Although this time it is on online mode, I hope in near future there would be an opportunity to visit the university and interact with the faculty and the students physically. So with that, so I would go and start my presentation. Let me share my screen. So it says host disabled participant screen sharing. Can you please give me a screen sharing? Okay, so Yeah. Am I audible and is my screen visible?
Yes, ma'am.
Okay. So very good morning to one and all delegates on the dais, all dear students and faculty members who are present today's meeting. So I'm going to speak to you today a very, very important topic, and that is how our food and production is challenged by the nutrition as well as the, the concern of the health. So as you all would agree with me, the impending changes that we are observing in the global climate, coupled with rapidly growing population, has resulted in challenges related to food and nutrition. On the other hand, we all have seen recent years, including COVID, that have shown increased frequency of high complexity diseases worldwide. Now, the pathogenic microbes, antimicrobial resistance, that is what we call as AMR, and diet play a pivotal role in infectious and lifestyle disorders. Plant and agricultural research is therefore continuously being challenged to elucidate the relationship between health, disease, and metabolism in one hand, and the effect of nutrition on the other. And these nutrient and immune response are very complex phenomena, and the exact physiological relevance and functional modification caused as a result of nutrient or anti-nutrient accumulation and also due to several types of diseases is very poorly understood. So now, if you try to see that, therefore, the challenges that has been put forward, even from the SDG goal, which we all globally have to attain by 2030, that clearly shows that there is a reduction in mortality that we have to achieve. We have to also achieve a reduced maternal mortality rate, child overweight and malnutrition, and eradicate malaria. Although it says eradicate malaria, in past 2 years we have seen it's very important to eradicate COVID-19, and I must tell you that infectious diseases are not only malaria and COVID-19, but there are many other infectious diseases which actually cripple the crop production in the field as well as cripple our human life and animal health. So therefore, these are very, very important parameters that need to be tackled needs immediate attention. So little bit, if I talk to you about the climate change and increased disease burden, this report came in 2017 from the Global Burden of Disease from the Institute of Health Metrics and Evaluation. If you actually go through it, the diet plays a pivotal role, and plant is actually gives 70% of diet and nutrition to human population, particularly in India. So therefore, looking to the plant's productivity, how we can make more nutritious food, how we can protect them from the pathogen infestation as well as many other abiotic stresses becomes very, very important issue. If we try to see the number of total YLDs and across the sex and by age group that has charted by the same document, it shows that from the birth to the 90+ years, because life expectancy has increased tremendously. However, how all different kind of diseases, whether it is a lifestyle disease or an infectious disease, is actually spread. So if you try to see the right hand side graph, it clearly shows you that the initial years of our life, we are actually threatened by kind of infectious disease as well as maternal and neonatal condition, nutritional deficiency. As we go along and mature enough, then it becomes more of kind of chronic diseases, which can be from infectious disease or NCDs, that is non-communicable disorders. So if that is the case, with this background in mind, my interest is always to find out how can we improve the plant health. How can we look for more nutritious food, particularly for protein nutrition, as well as a multi-host resistance? And we use a system approach to understand all these three facets. And today I'm going to talk to you, because of time constraint, I would be only focusing on to multi-host resistance. Now, when we come to the multi-host resistance in the agriculture field, it is a fungal infection which is enormous. And in that case, what we actually see in our agricultural field that has been shown, that there are the fungi which makes life problematic for not only plant but also human. If you just see this ProMed data which came up to the 2014 data that I have here, little old, but it's not much change has happened now also. This data clearly shows you, as the plant infecting fungi is gallopingly growing high, similar is the animal infecting fungi. Right hand side shows you global distribution of plant infecting fungi in the agricultural field, as well as the animal infecting fungi over in the globe. If you concentrate to the India where we all reside, you see in India we have more threat in the agriculture field rather than fungal infection in the animal or human. So therefore, it sets very important that we look for the mechanism by which we can plant can defend this fungal infection. How can we make the fungal tolerant crops? And now if I try to tell you, it has been told in a Royal Society paper by Fisher et al. In 2016 that global mortality due to fungal infection is exceeding that of malaria, which is a SDG mission, please remember, also breast cancer comparable to tuberculosis and HIV. So therefore, these fungal infections are actually not, I would say it's overlooked, but it needs immediate attention. If I try to understand how crop gets destroyed by fungi and how much perennial yield loss we get, it is more than 20% worldwide, with a further 10% loss in the post-harvest. Now, in that case, which are the major fungal diseases? There were another paper came up from Caroline Dean's group from John Innes, and that tells that there are 5 major fungal pathogens that we see: Magnococcal thoraciae, Botrytis cinerea, Puccinia graminis, Fusarium graminicerium, and Fusarium oxysporum. They top the list. And then major fungal diseases such as rice blast, wheat stem rust, corn smut challenge the global food supply, including Fusarium wilt in the chickpea. And this has been said by Sarah Gour, who is actually the chair in the food and food security at the University of Exeter in the UK. So in that case, I'm sure by now you are convinced how important to look into the fungal diseases worldwide. Now, this paper came in Science in 2018, and that shows that why this fungal species with reported global antifungal resistance is coming. Now, if you see from 1977 up to 2017, if you see left-hand panel shows the distribution worldwide of the fungal infestation in plant, right-hand panel shows you the fungal diseases in animal. If you see over this last 50 years, we have seen how tremendously these fungal diseases in agricultural field as well as and human is becoming more and more compared to what was a scenario in 1977. And therefore, there is a parallel driver of fungicide resistance, not only in the animal or human that we always call as AMR, but it is also very important in the field as well as in the clinic. Now, how this happens and how much death that we think will be attributable by AMR every year by 2050. If you see, it clearly tells that AMR is going to have a major challenge by 2050, where 10 million people will be under this threat. And it is the same thing in the agricultural field as well. And therefore, it's a very, very important area that the scientific discovery and scientific understanding must be strengthened. So how then, there is a concept all of us are hearing now, One Health. What is that? And how then human and plant health is connected? You know, we all inhabit on the soil, and therefore the soil health is the first thing which actually impaired the plant health as well as the animal health, both in the agriculture and the ecosystem. And as you could see, even the plant and animal health gives the nutrition, and that is a very, very important area. If we don't have a nutritious food, our immunity is lowered. You have seen in COVID-19 time, people are telling take more zinc, take more vitamin, and that means it gives you that how nutritive quality increase in food crop is very essential. At the same time, in the ecosystem service, environmental health, which includes the fungal resistance, becomes another very important area. And therefore, today we know that we cannot separate out plant from animal from human, and it makes a One Health concept, which is very, very important for global food security in the coming years to be there. And plants provide more than 95% of total food to human beings. Now, the subject that I'm going to take you further, I'm going to speak to you of host-specific immunity for a disease which is caused by a fungus, which is Fusarium oxysporum. Now, this fungus came only 50 million years ago in this earth, while human evolved somewhere between 180 million years ago, worm 350 million years ago, and chickpea, the crop that we work here, somewhere came in somewhere 280 million years ago. However, this particular Fusarium species can infect not only the plant, but also the animals. So we took to find out what is the commonality and the divergence that came in these two kingdoms, but where the pathogen still remains the same. So that is what we call as multi-host resistance. So now try to understanding. So let me now take you first to the plant, how this infection happens in the field. This fungus produces microconidia, and those microconidia goes through this root system and the collar region in the plant, and that makes a vascular necrosis, and that vascular necrosis ultimately makes the plant a problem for the water transport through the xylem vessel, and that yields to the drooping of the plant in the agricultural field, and that makes the plant not surviving. When we try to develop a model, because as you know, plant has an innate immune system, so as the C. elegans has also an innate immune system, which is a worm. And we wanted to know, because we were interested to understand the divergence and commonality between plant and animal, we quickly took this C. elegans as a model, established the model in the lab. And as you could see, the Fusarium oxysporum different strain, when actually assayed on this, we find that this strain, Fusarium oxysporum, that actually kills the Caesari. That means the oxysporum Caesari, this blue line, is actually giving the maximum threat even to the animal. So it is not only the species, but also at the pathotype level, it is the same pathotype which kills both the plant and animal. If you see that when you feed the worm with the Fusarium culture, you see normally the worm is surviving on the OP50, which is an equalized strain. But if you just deplete that and now you put them onto a medium containing the Fusarium, it can engulf the Fusarium. So it is a natural pathogen for the worm as well. And don't forget, worm also reside in our soil. So now if you see that, as we see in the plant, it is a vascular system. Similarly, in the animal, it is totally the digestive tract and the intestine that gets totally dissoluted when the worm is fed on this particular Fusarium species. So it is more or less the same path, although we call intestine, we call vascular system, but both have the common function, that is to transport the food and nutrient and the water in either in plant or in animal. So what we did, as I said to you that we are interested to understand everything from system approach, we actually tried to develop a spatiotemporal proteome and organ-specific transcriptome. We also combined a metabolite. So we actually did ITRAC based analysis, all phosphoprotein enrichment based analysis, and then what we found from all the system approach data, that in case of the the animal, here it is the worm, we find that in the worm, it has 6 chromosomes. And when you actually challenge it with and see the time course, you find that out of the 6 chromosomes, basically the chromosome 4 genes are totally downregulated, whereas rest of the other 5 chromosomes, the expression changes are actually more or less distributed. Now, we try to do a hierarchical clustering, which clearly shows in the right-hand panel to you that there are kind of blocks where you see the correlation, direct correlation between the highly overexpressed genes as in red, and in blue sectors represent the downregulated genes. Now we tried to enrich this, and the enrichment and we were interested in transcriptional and translational regulation of these pathways. So it shows that the TFs, transcription factors belonging to homeodomain ZF and hormone receptor family, were the major driver for the transcriptional regulation in worm. Now, we went ahead and made gene networks in the worm. And what this slide shows you, that we could find out that there are 6 modules. One is a cell fate and disease progression module, development regulators and immune response module. We have a lipid metabolism stress signaling module and a deregulated molecular machine organ patterning module, and also another module is homeostasis and co-signal regulatory control. Out of all these, we found that in the worm, the neurons and the intestine are 2 organs which are basically challenged because of this disease. And in that case, what we found 227 genes of neuronal origin and also 139 genes of intestinal origin in this worm, and which came very nicely. Now, very surprisingly, what we found, although there are so many genes showing differential expression, however, the DAF16, out of this neuronal 227 genes, 37 genes are the direct target of DAF16, which is a forkhead family transcription factor, as you all know, and 32 genes are the SERT2.1 regulated genes, and SERT2.1 is a histone deacetylase that all of you know. So therefore, we tried to then pick up the DAF16 and SERT2.1 to dissect out the global transcriptional change that's getting happening in the worm, and we believe that this is the pathway that possibly is important for the immunity against fusarium in the worm. Similarly, in the intestine, also we find out of 139, 21 and 32 are the targets of DAF16 and SERT2.1. So now, with that kind of a scenario, the next what we did, we actually found out the mutants of this worm, and we made a RNAi-based double mutant in the background of DAF16, and we downregulated because, as I said, in the worm, there are many neurons. Out of those, ASC and ASI neurons were known to have implication in the Pseudomonas bacterial infection. However, we are finding the neuronal genes, which I've shown to you in earlier slides, which are actually basically this GCY5, SRD1, GCY4, CH54. And in that case, we actually tried to now go and concentrate in the ASC and ASI neuronal genes, and which are important for regulation of innate immunity, and we think that is through intestinal DAF16. Why I'm saying this thing, as you could see from this graph of the killing assay, you find that all these things in a double mutant, you see if you make these genes mutated, the worms are actually survival goes down basically. So now, why we are saying it is intestinal DAF16, if you go back and try to see how many DAF16 genes are there in the worm, there are actually 6 DAF16 isoforms that we find there, out of which I showed you that intestine is most affected. So therefore, we posed the question, is the DAF16 isoform, which is present in the intestine, somehow is related to this immunity? Yes, indeed, that is what we find, that in a control worm, if you do not challenge it with the fusarium, the DAF16 is totally in the cytoplasm. However, if you have a DAF16 tagged with GFP, you find upon infection, the DAF16 moves from the cytoplasm to the nucleus. And therefore, we believe that DAF16 is a major regulator. To prove this thing, we did a % survival against a VP303 background. And in that case, indeed, we find that intestinal DAF16, that is a major driver of the immunity in case of, the worm that we find. So now in this case, what I'm showing, that the genetic analysis revealed that GCY5, which is an ASC function works upstream to the DAF16, whereas ASI specific SRD1 regulates the behavioral immunity. And that is where we found the pathway which is important for the animal to survive against this particular very, very pathogenic microbe that is the fungus. Fusarium. I'm now shifting to the plant, and then I will tell you how much is the common between plant and animal and how much is different between the two kingdoms. So if you know, chickpea is very important legume that we grow, not only we, but worldwide. India is a highest producer of chickpea. 64% of global chickpea is produced by India. However, what we find is that we still import chickpea from Canada and Australia. Why? That means our demand is not met by even producing 64% of chickpea in our country. Now, this chickpea is otherwise very much affected by fusarium wilt. And as you could see, these red triangles show you the fusarium affected area globally, and India is no exception. So now, if we try to see from a data from EqualSat bulletin that shows fusarium is a major challenge for chickpea production globally, 10% loss that happens if you compare to any of the other disease for chickpea. So that is where we try to then start understanding how this happens. Now, let me take you to a field which is naturally manifested by chickpea. As you could see, even there is a natural infection, you find there are some green patches and some yellow patch. So yellow patch are those chickpea variety which cannot tolerate and they become dead. However, the green ones can survive. So here, way back, we then posed a question. Does this mean that plant also has an individual immunity like we all human has? So this is where we try to understand host specific immunity. Just to tell you to the students that if you know the plant has innate immune response, that is governed by PTI and ETI, which are PAM triggered immunity and the effector triggered immunity. And in case of the host specific immunity, both the pathway would be working. So in that case, we took many of the chickpea cultivars and challenge them to understand the disease progression if there is any genotype and pathotype connection. And yes, indeed, that is what we found from our screening. And then we did a very elaborate physiological screening as well as biochemical screening. And as you could see from here, we picked up the most tolerant is WR315 cultivar, whereas the most susceptible is the JJ62 cultivar. From here, we then went ahead and developed global microarray based, as well as now we have the NGS based transcriptome. And from where we find that if you take the susceptible genotype in the left panel and you do this global expression change analysis and then go ahead and make the modular network, you find there are three major hubs that come up. One is for colonization, disease development, penetration, and small one are for perception. However, if you go and try to see what is happening in those green plants, you find three major hubs: ETI, related to ETI, PTI, and don't forget, this is a fungus. So not unlikely that we are getting a CTI, which is chitin triggered immunity hub. However, there are many genes which fall under these three commonly. So therefore, they're not specific to either of the CTI, ETI, and PTI. So we then went ahead and tried to ask what are those genes mostly in these hubs that we are seeing is highly deregulated. So these are few of such genes. And then what we did, we tried to see if you now challenge them, try to mimic the whole thing in the laboratory condition. What you find in case of the susceptible genotype, the pathogen load increases tremendously, what we find from the analysis of the fungal DNA overload. And in that case, what we see that it is what actually giving the protection at the first level is nothing but the ROS. And this ROS is, if you could see in the resistant genotype, you see a very huge ROS burst in very early of the infection, that is from the 6 hour, and between 6 to 24 hours, a plant is capable to actually make the pathogen not to enter it. So it is at the first level of protection, and that is the cell wall. So with that, there is emerging role of different organelles in different cellular process, not only in plant, but in also human. We are working for many years using proteomics to dissect the role of organelles in different cellular process. I'm going to speak to you today, these two papers, which is proteomics and journal of plant journal paper. And in that, what we have found is that when the pathogen go inside, you see the vasculature change in the vasculature, you see the infestation of the fungus up to 240 hours. And then what we found that if you do a scanning electron microscope, the cell wall basically is totally dissoluted in case of the susceptible variety. So then the question was, if the chitin triggered immunity hub is coming up, it has been shown that the deacetylated form of the chitin is actually does not become a PAM, but in contrary, it gives a protection. And many people have used chitosan nanoparticles in the agricultural field. So we also tried to see the effect of chitosan on tick seedling challenged with fusarium. So you have the control fusarium treated, control fusarium untreated for both JG62 and WR. And what we could see that yes, indeed, when you actually use the pre-treatment with chitosan, that gives you more than 60 to 70% protection. So, as I said, we are interested in understanding the organellar role in immunity. So, in this, I'm just going to talk to you about the ECM. So, ECM is the outside. And as you saw that there is a direct morphophysiological manifestation, we went ahead and purified all the ECM proteins, and we did different kind of assay in order to ensure that our ECM preparation is at least 99% homogeneous and pure. So with that kind of thing, we then went ahead and did ultrastructure analysis and carbohydrate composition analysis. This slide tells you that it is the pectin, which actually is a major carbohydrate, which is related to the wall strengthening in the immune species. Now, with that, we went ahead and did a DCMS based and LCMS based metabolite analysis, and very clearly what you find is that it shows such a nice similar profile when you have a susceptible genotype, and now you have actually treated the susceptible genotype with the chitosan, that reverts back to all the metabolite profile just as a immune plant, that is WR. So the pathways that got enriched are galactose metabolism, amino acid tRNA metabolism, glucose, methane, and some of the amino acid metabolic pathways. Now, we then went ahead and developed the ECM physiosome in the susceptible variety, where you find wall biogenesis, redox homeostasis, wall architecture remodeling, and disease response are the major process driver, whereas germine APX2 are the major protein that we believe are very much involved in susceptibility. When you see the chitosan regulated ECM immunome, in that case, in the same variety, now you are seeing a totally different kind of biological processes which are involved with cell wall reorganization, wall acidification, glycan degradation, hydration, acidification, EATP signaling, Ross homeostasis, and so on and so forth. So from here, we then tried to ask the question, if you now challenge this with the susceptible genotype with chitosan, do you see a reversal in the Ross? Yes, indeed, that is what the lower panel is showing you. And then we measured all the Ross related enzyme. What this slide tells you is the APX activity, SOD activity, LOX activity, glutathione peroxidase activity, all were much more in the chitosan treated plants. We went ahead and tried to see, because in the literature it has been shown that maybe this, and in our hub we got a EATP signaling. So this extracellular ATP signaling possibly is a major first line of defense that the plant has with the Ross in case of the fusarium. So it was shown in the literature, ANO also regulates EATP signaling, which may be, we think, linked to stomatal function during fusarium attack. And in that case, we saw the opening and closing of stomata, as well as the stomatal contact extent, transpiration, ANO measurement, and EATP measurement. Indeed, we could find that chitosan-induced ANO particularly regulates the EATP signaling, which is then linked to the stomatal function during this fusarium attack in the immune situation. So this is the final mechanistic detail with which we come up with the chitosan-induced CTI during fusariosis. This is the first report globally of the chitosan activity during the CTI. All the people have worked more in the chitin, but chitosan was overlooked all these years. So with that, we then picked up from all this network analysis, one of the major transcription factor, which we named as IRF817. It is a BHLH domain containing factor. We have now patent across globe on this IRF1, and we went ahead and did many of the things. I'm going to show you final result, although we now know which protein it interacts with, how this protein's promoter is regulated, how this protein regulates what are its downstream targets. However, the final result that we found out, if you now overexpresses IRF1, it actually imparts the tolerance in the overexpression line, and it makes the plant more susceptible in the RNAi line. So this conclusively proves IRF1 is a major regulator of immunity, unlike to DAF16 in the animal. So to sum up, the longitudinal spatiotemporal multiomics analysis from our studies and from the derived biomolecular network revealed organ and organelle function in diverse kingdom during Fusarium disease. As I have shown you that they are highly conserved regulatory components and pathways, and very interestingly, in case of the plant, beta-7 transmembrane receptor, LRR protein, serpentine receptor that regulate the signaling event in worm, but in plant it is a folate and glutamate receptor. When you go and do a closer inspection in the plant, it is the oligosaccharide signaling, whereas in case of the animal, it is basically the sphingolipid signaling through G protein pattern sensory neuronal pathway. And then finally, we could find out the novel insight on cross-species immune signaling that might impinge upon the surveillance mechanism of innate immunity in this very important multi-host pathogen response. And this we are now trying to take for kind of gene edited lines to link our basic findings with the translational output. And IRF1 is definitely the major regulator of plant immunity. With that, I would end my talk. I'm just here to speak in front of you. All this nice work have been done in over the years by all my very, very talented graduate students, postdocs, and other GRFs and SRFs in the field. These are different time frame, the students that I had, and current the people who are involved in this work was involved and still are involved. From the current lab members, Konika, Rajul, Archana, Shobha is just joined last year, and then the former lab members are Nagaraju, Nashiman, Swaraj, Shudeep, Pooja, Arunima, and Diman. All this work have been are the contribution from their work, and it means I must acknowledge the funding without which nothing could have gone better, and that is from DBT, NIPGR, ICMR, DST, SCRB. So with that, thank you again, and I can take very quickly one question, not more than that, because I have to be online for a very important IFD presentation to DBT right now, and it's already on. Thank you.
Okay, thank you, Professor Chakrabarti, for giving a great lecture to us, to enlightening our audience. Actually, our students and teachers were learned a lot from you, from your comparative work between comparative genomics of C. elegans and plants in response to, infection of fungus, which tells us the commonality between the defense responses of plants and animals, and mostly that centers around reactive oxygen species. With these words, because Professor Chakrabarti has to go for immediately another meeting at 11:15, she told me before. She had a meeting with Department of Biotechnology. We thank you, madam, very much for your kind presence here. And we are not going to have a question and answer session right now because it's difficult to control one question right now. Okay. So with these words, I thank you. Thank you very much, madam.
Thank you, sir. Maybe later I would have chance to interact with students more. Thank you very much, and wish you a very successful 2-day intense scientific discussion. And I'm sure from these discussions, students will be benefited enormously.
Okay, thank you. Thank you. Okay.
कहाँ से बंद करें?
Now I once again request all the dignitaries to please take their place at the dais. I would now request Ms. Lavanya to enchant us with Saraswati Vandana.
या कुन्देन्दु तुषार हारधवला या शुभ्रवस्त्रावृता या वीणावरदण्ड मण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहाम् वरदे वरदे हे वरदे वीणावादने वरदे वरदे हे वरदे प्रिय स्वतंत्र रव अमृत मंत्र नव भारत में भर दे वरदे हे वरदे वीणा वादनी वरदे नवगति नवलय ताल छंद नव नवल कंठ नव जलद मंत्र नव 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 के नव विहग वृंद को नव पर नव स्वर दे वरदे वरदे वीणा वादनी वरदे काठ अंधुर के बंधन स्तर काठ अंधुर के बंधन स्तर महाजननी जो तिरमय निरझर कलुष भेद तव हर प्रकाश भर जगमग जग कर दे वरदे वरदे वीणा वादनी वरदे थैंक यू
Thank you, Lavanya, for such beautiful recitation. Now, taking this event further, I would request Dean Sir to kindly felicitate Honorable Vice Chancellor Sir. I would also request Professor B.C. Tripathi Sir to kindly felicitate Dean Sir. I would now like to invite Honorable Vice Chancellor Sir for the welcome address.
Thank you. Chief guest of today's event, Dr. Subra Chakraborty. Of course, she has finished her session. Professor Parmanand, Dean Academics and Dean of the School of Engineering, and Tripathi ji, Professor Tripathi, the eminent scientist, Professor, HOD, other faculty members, and my dear students. Very good morning and congratulations for organizing such an important event, Cellular and Biomolecular Technology. It will be a two-day event involving many experts who will be presenting their works, the research trends, and other statistical inputs, data, which will make this two-day event very fruitful. And I'm sure that faculty members and students will acquire and they will be benefited by the content which will be displayed and delivered in this event. Sarda University is now on priority based focusing the research, the entire spectrum of biotechnology, including life science, because this university has no medical schools, dental, nursing, pharmacy, life science, biotechnology, agriculture, etc. And recently we have seen that very important projects, funded projects have been brought by the researcher in different departments. And also we have been able to establish a few state-of-the-art research facilities through funded project as well as through the funds given by the university. So university is now planning to establish one Sarda Research and Innovation Center in a separate building, and that building will be constructed very fast, maybe within 1.5 years. And basically the research will start with biotechnology domain, because specifically with the experience of pandemic, whole civilizations has realized that what kind of importance and attentions to be given in the biotechnological sector. It's biology as well as technological solutions. And not only that, along with that, our environmental science also will be equally important, because despite having all these technological supports, devices, facilities, the most important thing is the life matters to survive on this planet. So health is wealth. If our health is all right, and if we are able to fight different viruses and diseases and make the people very healthy, then they can contribute towards the society. And also we can make the society free from any threats of such virus or any other diseases. And already we all know that the way India has handled the COVID. Last week we have completed 100 crores vaccinations, and whole world is astonished, and they are saying towards the India how just it was possible. Because you know that we are almost 1.4 billion people on this planet, and every 6 persons on this earth is Indian. So it's not that matter of 100 crore populations have been vaccinated. In terms of the number of countries now you can calculate. It may be entire Europe, or it may be entire American or African countries. So you can take one of such big continental have been vaccinated. So, and that too, when pandemic happened, we know the preparedness available with India. And from that scratch pad, zero level, after the first wave, we have seen the experience in the second wave, where also the preparation was not much because of time constraint. And also India is known to be very biomedically, the medical support system is not much well equipped because of a very vast country, because India is a very vast country and huge population. But with a very short span of time, this immune vaccination, 100 crore people, and then suppressing the probability of rising the third wave itself is a great achievement. So whole nation should be very much proud. And these are all contributions coming from the all biotechnological scientists, life science people, doctors, and many more strategic developers and planners are also involved. Because such a huge country, the distribution of vaccine and its maintenance of the status, up-to-date status, and making all these vaccines available in different parts of the country, including interior. You'll be astonished to hear that in the interior to interior villages, these vaccinations was carried out very meticulously with top priority, and there in fact percentage was much higher. So if civilization is to survive, the planet is to survive, and if planet is surviving, the first thing, the life is to survive. And life will survive if really with such kind of research, we provide all these medical facilities and able to sustain even during such pandemic. So all these molecular and biomolecular level in-depth research is happening to understand the behavior and also their influence and their life cycles, and thereby finding out the facts and reasons and coming out with certain solutions which we can fight at the molecular level, specifically in the biomolecular section. I'm sure that the actual vaccine, this is probably what to the best of my knowledge, it is the immune boosting vaccine. And maybe one vaccine you need to take for your entire life, like other vaccinations, smallpox vaccinations, other vaccinations, yet to will come very shortly, and then whole world will be totally free from such threats of any kind of pandemic in future. So a great event organized in the relevant time, and I wish you all the best and grand success of this today event. Jai Hind.
I thanks VC sir for his profound vision, grateful thoughts and encouragement. Taking this event further, I would now like to invite Professor Permanand sir, Dean Academics and Dean SET, to deliver the introductory address.
Very good morning to all of you. Honorable Vice Chancellor sir, Chief Guest Dr. Chakravarti Madam, Distinguished Professor B.C. Tripathi sir, HOD Dr. Abhimanyu Jha, dear faculties and our dear student members. Good to know that Department of Biotechnology has organized a national conference on cellular, molecular and molecular technology. I have seen in the last few days the way the people were conversed together to make this conference successful, and this is what we can see here. The conference is started with good note. Conferences are being organized to discuss the research papers, whatever latest recent research has been going on, people are discussing and the people are learning from each other. Of course, due to the pandemic's COVID-19 situation, this conference is in a blended mode. Otherwise, such conferences should have been in a physical mode. As rightly said by Honorable Vice Chancellor, sir, biotechnology is one of the major field to be taken in the future. Rightly said and shown by the chief guest, the vision of 2030, where she has addressed the requirement of different kind of requirement in the field of biotechnology and the life sciences. Of course, in the Department of Biotechnology, people are working on the plant technology, animal technology, as well as on transgenic. Some of you are working on the stem cell technology. You know that the stem cell technology is the need of hour. In the COVID-19 situation, we have seen the shortages of different kind of medicines and other supported materials. We need to converge together. We need to go for the multi-departmental research. Sada University is unique in itself, having the hospital, dental, nursing, and allied health sciences. Over the period of time, we are not able to see many number of collaborative research among these schools. There are many fields which are still to be explored, like your oncology, cancer, Alzheimer, where the research is still going on, but is still a lot to go. With this, I like to thank you all the organizers, the team of the head of biotechnology, for organizing such a wonderful national conference under the leadership of Professor B.C. Tripathi sir. Thank you so much, and I hope in these 2 days everybody is going to learn a lot, many things. Thank you. Thank you.
I thank Dean Sir for his kind words, remarkable thoughts, and unwavering support. Now I would like to invite Professor Abhimanyu Jha for sharing few words with us.
A very warm good morning to all of you, Honorable Vice Chancellor Sir, Dean Sir, Professor B.C. Tripathi Sir, and my colleagues as well as my young students. This conference was planned in a very short span of time on a very contemporary issue, although this is a broader issue of cellular and molecular biology. This conference would not have been possible without the motivational and long-term vision of our Honorable VC, Professor Sebraham Khara sir, as well as the inspiring leadership of Dean School of Engineering and Technology, Professor Permanent sir. We were able to gather and bring under one umbrella so many renowned speakers from many prominent universities as well as prominent institutes across the country because of Professor B.C. Tripathi sir. He was very instrumental in bringing all the resource persons under one umbrella for this national conference. I would also like to thank Dr. Saurabh, the co-convener of this conference, for playing a very instrumental role in the smooth conduct of this conference. And I would also like to thank, before telling you about the details of the conference, I would also like to thank all my faculty members, Professor Tanija, Dr. Sachin Tewatia, Dr. Amit, Dr. Monica, Dr. Garima, Professor MC Pandey, Professor Peetam Chandra sir, as well as Dr. Neeraj, Ms. Priya, Dr. Amit Kumar, and Dr. Bhupinder Kaur for working so nice as a team. And we have been able to bring you into a very good contemporary topic. And you are with us for 2 days. And for the 2 days, we are going to have so many technical sessions. We are going to have very important talks, and you're going to learn a lot. I would like to tell you one thing. There was a saying by Theodosius Dobzhansky, a very renowned evolutionary biologist. He said, nothing in biology makes sense except in the light of evolution. I would like to add a point here. Nothing in biology makes sense except the knowledge of cells and molecules, because cells are the structural and functional units of life. And as you all know, biology is the study of life. Molecules, if they are not present, we cannot imagine the cells. And if the cells are not present, we cannot imagine the life. In this recent trying and testing times of COVID pandemic, biotechnology proved that it is a boon for the mankind. And as rightly said by VC sir and Dean sir, biotechnology has been a major force which has helped the mankind survive during this time of COVID-19. We would like you to interact with the speakers. We would like all the students to come forward, ask questions, because doubts are the basis of science. Without doubts, the science will not progress. So you need to be inquisitive, come forward, be enthusiastic, and let's learn a lot from all the speakers. I would like to thank you all for this event, and I would like to request you to be patient listeners and enthusiastic learners. Thank you. Thanks a lot.
Thank you so much, sir, for your motivational words. We will now have a very short tea break for 15 minutes. After then, we reassemble and take further. Thank you.
अरे यार
Hello. Oh, okay.
I request Professor to enlighten us with his talk.
Okay. Am I audible to people? Hello? 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 Am I audible?
Yeah, sure.
Okay.
You are audible for you, sure. It happened.
Uh,
उसको क्या?
Can I share my screen? How do I do that here?
इसको ठीक करो.
Screen.
Sir, please wait for a moment.
Okay.
तुम सर के साथ क्या चाहिए प्लीज.
Yes, now you can start, Ramesh sir.
Okay. Whether my screen is
Visible. Yes, screen is visible.
visible? Yeah,
Yes, yes.
okay. Right. Okay, thank you for this opportunity to ask me, to give a talk to Strada University. For the first time, I'm giving it to this university. I hope it would be useful. Over a period of time, we've been working actually on translational regulation. Hello, can you hear? Hello.
Now all of you leave.
नहीं करेंगे चलिए।
Hello. Hello.
Yes, Professor, you are audible.
Okay. My screen is visible and I'm audible, right?
Yes, sir.
Yes.
Okay. Now let me start. This has been a long time. I've been working actually on translational regulation of protein biosynthesis regulation. That is what I do not know. A lot of times I feel this area is not given much of an importance, in the sense most of the people work on transcription and they're happy to see an RNA, but they would not like to see a protein being made out of RNA or not. And if you look at it, actually the translational controls are not fantasy anymore, they are facts. And a lot of times, unless the protein is there, you can't assess the function of it. And quite often people say that they have a gene in hand, but they may not be having a protein or an antibody to really work it. And there are many regulations, actually, the eukaryotic protein biosynthesis. If you look at it, there are several control mechanisms that turn on and off the protein synthesis as and when it is required. And one of the regulatory elements is the phosphorylation of EIF2 also. And that is what we've been working on. And my talk, actually, I'm planning to divide it into describing the initiation step in protein biosynthesis in eukaryotes, heme deficient lysates, particularly with which we worked. And in fact, they contributed quite a bit to understanding the protein biosynthesis. And not only heme deficient lysates, but other cell-free translational systems as well, like germ systems and HeLa systems, etc. And then I'll discuss about what is EIF2 and the subunits there, and its recycling by EIF2B and the kinases that phosphorylate EIF2 and the mechanism of phosphorylation of EIF2. How does that inhibit the protein biosynthesis? And then how is that connected to integrated stress response and unfolded protein response, which are major factors actually to control several metabolic and neurological disorders. And then the importance of EIF2 phosphorylation regulating the virus infection. And in that context, I specifically focus on a couple of proteins that we work with, like the baculoviral proteins that inhibit host cell EIF2 phosphorylation. And then the importance of EIF2 phosphorylation in aging, particularly what happens, but the aged animals we found, actually they are unable to mount unfolded protein response or integrated stress response, which is actually an adaptive signaling pathway. And that may be the reason for many of the metabolic and neurological disorders that the aged people suffer from. And then we also recently been working on mitochondrial dysfunction. How is that connected to EIF2 phosphorylation and ISR activation? So these are the things that I plan to cover, and let us see if I can do that. Or I'll quickly go through most of the time the summaries rather than going into the details of things. See, in the protein synthesis initiation, if you look at it, there are several factors. You see, this is only the initiation step. Imagine in a cell, you have so many genes and so many proteins, and the number of interactions really staggering. And in fact, the function, physiology, metabolism, health, and disease of cells and cell types, if you look at it, they're regulated by multiple interactions, not one. In fact, if you're a biologist, you will go definitely a philosopher at the end of the day, because what defines the outcome, be that physiology, metabolism, health, and disease of cells and cell types, although the genes are the same, but interestingly, it is the interacting partners that define mostly the outcome. Here at the initiation step, to start with, the importance of EIF2, which is called eukaryotic initiation factor 2, its basic function is it delivers the initiated tRNA to the 40S ribosomal subunits. And in a GTP-dependent manner, it requires GTP for it to bind the initiated tRNA, that is a methynyl tRNA initiated tRNA, and form a ternary complex. Then it is joined by at least 4 or 5 of the initiation factors, EIF1, 1A, 3, and 5, and it forms a multi-factor complex that goes and binds to the 40S ribosomal subunit and becomes 43S complex. Now this 43S complex then joins the messenger RNA. The eukaryotic messenger RNA, as you know, has a 5' cap and a 3' poly A tail, and the 5' cap is bound by a complex of proteins EIF4F, consisting of 4E, 4G, and 4A, that are important not only for binding to the cap and also to believing if there is any structure in the messenger RNA during the initiation. These helicages like protein, like 4A and 4B and H, they unwind the RNA structure. And the 4G actually is the scaffolding protein. On one side, it interacts with 4E, and another side, it interacts with the poly A binding protein that is bound at the 3' end of poly A tail. And this 4G now interacts actually the EIF3 of 40S complex, and then it becomes a 48S complex. And then this 48S complex looks for the start A UG codon, and it scans the messenger RNA and finally identifies the start codon and puts the tRNA there. Before it does that, I mean, it really reaches to the start codon, then the 60S subunit joins. And in this process, actually, the EIF5, which is having a GTPase stimulating activity, stimulates the GTPase activity of the gamma subunit present in EIF2. That has a GTPase activity. And then the GTP bound to EIF2 is hydrolyzed, and it is released as EIF2 GDP, a binary complex. And this is generally non-functional unless it is exchanged by GTP. And EIF2 GDP has a better affinity for EIF2 in physiological conditions in the presence of vignesia. So this exchange reaction cannot take place by mass reactions, but it requires a catalytic factor like EIF2B, which exchanges, actually it is a guanine nucleotide exchange factor that exchanges the GDP bound to EIF2 for GTP. Then it gets again into another round of initiation cycle. So this is the basic protein synthesis and the role of EIF2 there. And that is what people found out. And very early days, actually, when people were studying protein synthesis in reticulocyte lysates or gravid reticulocyte lysates, particularly the red blood cells, what I'm talking about. And whenever there is a heme deficiency, then there is an inhibition in protein synthesis, because mostly, imagine, the red blood cells have the globin messenger RNA. The globin messenger RNA translation doesn't take place in the absence of heme. So heme shuts down. This is an iron protoporphyrin compound, actually. And in the presence of heme, these lysates do a good job of protein synthesis, and that is measured in terms of C14 leucine incorporation. So these are test tube reactions where you have the red blood cell lysate, and the lysate is having messenger RNA whose translation you are monitoring using the C14 leucine radio-labeled amino acid. So the incorporation of radio-labeled amino acid into the protein will be looked into, and then just one minute. Sorry. And then in this scenario, what happens is if you add delayed addition of heme to shut off lysate, then it resumes protein synthesis. That is the one which people wanted to identify what happens at this junction when, in the absence of heme, how the protein synthesis is shut down. People looked at it and they found actually it is due to phosphorylation of EIF2. And the phosphorylation of EIF2, and when you add delayed addition of heme, that is, once the protein synthesis is inhibited, if you add heme in, then the restoration of the protein synthesis occurs, because in that process, heme actually inhibits the kinase, and there is an endogenous phosphatase that dephosphorylates this EIF2 alpha P and restores protein synthesis. But then people observed actually the phosphorylation part. You see, a lot of us, we study phosphorylation, but we still do not really realize how much of phosphorylation, for example, is going to affect, hello? How much of phosphorylation is going to affect the real function. In this case, not the function here, 30% of EIF2 phosphorylation is sufficient to inhibit the protein synthesis, although 70% of EIF2 is unphosphorylated. So that was the question, how come only 30% is inhibiting? Then people look for if there are any more factors, and they realize that there is yet another factor that is called EIF2B, which actually exchanges the GDP bound for EIF2 with the GDP. But actually, when you assay for the activity in vitro or in test tube reactions, people use labeled GDP, and then they study the exchange of it with unlabeled GDP. And that gives actually an idea. If EIF2 is not phosphorylated, then you can see the exchange of GDP bound to EIF2 can be quickly displaced by unlabeled GDP or GTP. But if it is phosphorylated, then it doesn't happen. And then what happens? Actually, what is the level of 2B that people looked into? And the level of 2B is only 30% of total EIF2. If there are 100 molecules of EIF2, there is only 30 molecules. So that is the rate limiting factor. So whenever 30% of EIF2 phosphorylation occurs, then what we suggested then was all the EIF2B, whatever is available, must be getting sequestered and forming a complex. So you get an EIF2 alpha T2B complex, in which actually the 2B will not be able to do this GTP exchange any further. So it gets complex with phosphorylated EIF2. That is the story here. And then with that, you see, we started also working on that problem. Later, I'll come to that. In fact, the importance of cell-free translation systems, there are plenty of them. Initially, people used to decipher the genetic code, and then they tried it with hello. Hello. Is there anything? Okay. So one of the some of the importance of potentialities of cell-free translation systems. I'm just telling because most of you are also biotechnology students, and a lot of times you may not have used even. The cell-free translation system is not there in the practicals. And so here, what people earlier have used actually cell-free translation systems coming from E. coli to decipher the genetic code, and also to determine the role of eukaryotic cell-free translation systems were used to understand the importance of signal recognition particle and docking protein in the regulation of secretory protein biosynthesis, and also to determine the pre-pro sequences in proteins, because most of the time the proteins are having pre-pro sequences which are eliminated to get the right level of protein. I mean, the protein which has lost some of the unwanted amino acids so that it gets its structure better. So the pre-pro sequences are always lost actually in the cell because of the proteases. They're like a chymotrypsinogen becomes a chymotrypsin. And to really see what is the full-length protein actually that is being made from a messenger RNA, the cell-free translation system devoid of the proteases could be very useful. and also to determine the inhibitory steps in protein synthesis by antibiotics, at what level they inhibit, at initiation level, elongation level, or termination level. And then the reconstituted cell-free translation systems were used to identify defective translational components like ribosomes, initiation, elongation, and termination factors. And we also used them to evaluate the mechanism of action of novel compounds or small inhibitory or activator molecules in translation. And also they could be used, the so-called hybrid arrest translation systems. One can also use it to pick up a clone which carries the gene of your interest if you have the right mRNA. So the mRNA translation can be done. And if you add the gene of your interest, then you see the translation is shut down because of the base pair complementarity. So you can actually pick up from a library a clone that harbors the gene of your interest using reconstituted cell-free translation systems. And previously, of course, we have used this system, the cell-free translation systems, to study some of the novel compounds that are being made, like vanadate and vanadyl complexes, pyrroloquinol and phenol, on how does that affect actually the protein biosynthesis and other things. And a quick glance of EIF2 subunit functions, if you look at it, there are 3 subunits. It's the heterotrimeric, the alpha subunit, beta subunit, and gamma subunit. And the alpha subunit is implicated in the regulation of protein synthesis. It has one phosphorylation site and gets phosphorylated on the 51 serine residue, and it is phosphorylated by multiple EIF2 alpha kinases. And the beta subunit is a hub for protein-protein interactions. And it also has several phosphorylation sites, but believe it or not, we have shown that, but still we do not understand the physiological significance of the phosphorylation. It can be phosphorylated by CK2, PKC, PKA, and DNA-dependent protein kinase. It also interacts with mRNA, whereas the gamma subunit, which is structurally and functionally related to the bacterial elongation factor TU, or elongation factor 1A. It is the last subunit, and this one has, it binds to GTP, GDP binds to mtRNA, and also it has the GTPase activity. So for all functions, actually, the gamma subunit is important, beta subunit aids that, and the gamma subunit and alpha subunit is implicated in the regulation of protein synthesis. And there are several kinases which are discovered. To start with, people have discovered the EIF2 alpha kinase in heme deficient lysis. What exactly the kinase which is phosphorylating EIF2 in heme deficient lysis? Then it turned out to be heme regulated kinase, which they thought is mostly confined to erythroid cells. But now actually it is also present in tumor cells. That is what at the end I will come to that. But the heme-regulated kinase is one, and then people started looking for other potential EIF2 alpha kinases. Then, whenever there is a virus infection, particularly double-stranded RNA virus, the PKR gets activated, and PKR phosphorylates also EIF2. And subsequently, there is a third kinase which is discovered, that is the GCN2 kinase. And this GCN2 kinase gets activated in the absence of amino acids, particularly when uncharged tRNAs are accumulated in the cell. They can bind to a moiety here, and then they can activate the GCN2 kinase. And then recently, about 10 years ago, this PERK kinase has come, or pancreatic EIF2 alpha kinase, or endoplasmic reticulum resident EIF2 alpha kinase. This is membrane-bound kinase, and this gets activated in response to unfolded proteins. So if you look at the PKR kinase structure, actually the PKR kinase has double-stranded RNA binding domains, and HRI kinase has heme binding domains here and here, and PERK has a cytoplasmic domain and luminal domain, and this is the one which actually binds to unfolded proteins and gets activated. And GCN2 kinase has a uncharged tRNA binding site. Although they all phosphorylate the same serine residue on EIF2 alpha, their regulation are different. One requires double-stranded RNA, one requires heme, another requires unfolded proteins, another requires RNA acid starvation or uncharged DR. But these 4 kinases are also implicated actually so far in other stresses in like salt stress, for example, you take oxidative stress and other things. One of these kinases are getting triggered so far. These are the 4 well-characterized kinases that are seen. And we were actually trying to understand the mechanism of protein synthesis inhibition caused by AR2 alpha, and we postulated that there must be a complex formation between EIF2 and EIF2B, and this is a complex which is now considered to be a 15S complex in which EIF2B becomes non-functional. And to doing that, we have made several mutations. One is a wild type EIF2 alpha, and then the adjacent to the 51 site, there is another 48 serine residue that we have modified to alanine. And then if you take the S51 and modify that to alanine also, and then the serine is replaced by an aspartic acid, then it becomes S51D. And now this one actually phosphorylation of serine 51 residue inhibits protein synthesis, inhibits EIF2B complex or formation 15S complex. And S48A, although in this case, the wild type phosphorylation like S51 phosphorylation is seen because S51 is not mutated, but it is the adjacent serine which is modified, and it is not phosphorylated. Only one phosphorylation site is there in the IF2, that is serine 51 site. But if you modify this adjacent serine to alanine, then what happens, although this is phosphorylated, then you don't see actually inhibition protein synthesis of 2B activity. So we presume that probably whenever 51 phosphorylation occurs, then for this to interact with the EIF2B, a tight interaction is required, and that tight interaction is promoted by the serine 48. And if that is replaced by alanine, then you don't see a tight interaction between phosphorylated EIF2 and 2. And also S51D behaves almost like wild type phosphorylation. It is a phosphomimetic form of EIF2. And the 2B also we try to characterize, and the 2B has 5 subunits to start with. It is a heteropentameric, but now it is considered to be heterodecameric. Without, of course, I'll come to that later. And it has 2 of the subunits. They are catalytic subunits. The gamma and epsilon ones are catalytic, and the regulatory subunits are beta, alpha, and gamma. So the catalytic subunits, they exchange the GDP from EIF2 for GTP, and the regulatory complex is the one which tightly interacts with phosphorylated EIF2. And that is how it is going to inhibit actually the function of 2B. So in a EIF2, which is bound with GDP with alpha, beta, and various physiological conditions, what you see is actually EIF2 is phosphorylated, and the phosphorylated one binds to 2B, and where it forms a complex, in which condition actually it is an inactive complex, or EIF2B becomes non-functional. But then, in the absence of phosphorylation, it can exchange the GDP for GTP and then joins the methRNA. And that is how we get the ternary complex formation that gets into the initiation cycle of protein synthesis. And we also used expressed actually all the other subunits, alpha, beta, and gamma, using baculovirus expression system. And we studied the protein-protein interactions and also studied the phosphorylation of the beta subunit, and it is phosphorylated by various kinases. But we haven't, as I said, really gotten into any functional aspects of it. But all the 3 subunits of EIF2 are expressed, and beta and gamma actually they co-migrate together a lot of times. Although the molecular masses are different, but because of some residues, amino acid residues, their molecular mass appears to be more or less the same. And then we also observed alpha subunit is known actually is a substrate for caspases. And they have shown caspase 3 can play. When we express beta subunit, we also found beta subunit could also be actually processed by caspases. And not only that, the phosphorylated forms of alpha and beta, we find actually they're resistant to caspase activity. That is what we have seen. And phosphorylation of beta subunit by various kinases, and we try to look at it. Actually, what happens, there's the ability to recognize a caspase. Phosphorylated forms are not cleaved by the caspase, whereas the non-phosphorylated forms are cleaved. And as I was mentioning, actually, how phosphorylation recently, Pinky Ramakrishnan lab has given actually some idea about using the crystal structure of EIF to alpha P2B together. They said that through direct interaction of phosphate groups with residues in EIF2B alpha, this is the regulatory subunit, one of the regulatory subunits of EIF2B, actually can interact with the phosphorylated EIF2 alpha. And indirectly, by inducing contact, you see, EIF2B, as I said, is now considered to be a heterodecameric, and on either side you find EIF2 binding, and the alpha subunit actually binds to the one of the 2B subunits alpha and also to the delta. This is the regulatory one, and using the D1 helix structure. And whenever it is phosphorylated, there is a reorientation of these amino acids and also the phosphate actually that facilitates a tight interaction between them. That is how the 2B activity will be lost. That is the explanation they have given. And also there is another protein which is also important, EIF4-EBP initiation. EIF4-EBP is a EIF4E binding protein. Actually, it binds to EIF4E. BP, and in which case EIF4E cannot actually interact with other partners which are important for its function. But in whenever there are growth factors, nutrients, and other things are there in the cell, and the cell is actively doing protein synthesis, then EIF4E BP gets phosphorylated. Once it is phosphorylated, then actually it comes out of EIF4E and facilitates the EIF4E to interact with the multifactor complex and start resuming the translation. So the 4EBP1 phosphorylation is almost opposite to what you see EIF2 phosphorylation. While the EIF2 phosphorylation downregulates protein synthesis, EIF4EBP phosphorylation upregulates protein synthesis, particularly the cap-dependent translation. Now, what is integrated stress response? So people once they discovered actually EIF2 alpha, and they have discovered several kinases, and then they have discovered several stresses actually are going to promote EIF2 phosphorylation. And they looked at it, no doubt initially people found the translation was getting attenuated, but at the same time in whole cells you see there is a gene-specific mRNA transcriptional induction is going on. There are some gene-specific mRNAs which are translated very efficiently actually in response to EIF2 phosphorylation. While most of the general mRNAs are shut down, some of these other mRNAs, they're expressed. And what are these mRNAs? Two of them found to be very important: ADF4, activated transcription factor 4, CHOP, which is a CEBP homologous protein. And one promotes actually the survival and the other promotes the death. And also you get GAD34. There are many transcripts later they have recognized actually. You see, the normal mRNA translation is shut down, but these ADF4, CHOP, and GAD34, what is the speciality of these mRNAs? They have many upstream open reading frames preceding the main ORF. And this is the general structure of these mRNAs, and all these mRNAs are translated wherever there are upstream open reading frame, much better than the other mRNAs whenever EIF2 alpha is phosphorylated. When ATF4 is implicated once it is made, it in turn actually allows the expression of some of the autophagy genes as well as genes involved in redox metabolism and chaperones, and that is how it promotes cell survival. Whereas the CHOP actually promotes death-inducing proteins, and GAD34 is a cofactor of EIF2 alpha phosphatase. You see, GAD34 is a phosphatase that dephosphorylates EIF2 alpha and facilitates the resumption of protein synthesis. But whenever the resumption of protein synthesis occurs before actually the stress is relieved, then there will be more oxidative condition generated because there is excessive protein synthesis in the cell, which is beyond the capacity of the protein folding, and that can result also in accumulation of unfolded proteins. So that is how GAD34 could be good and bad, particularly if it is activated and starts resuming protein synthesis when the cells are still experiencing stress. It will be a kind of death signal for them. And then came, actually, as I was mentioning, POC was discovered. POC is a kinase which is found in the endoplasmic reticulum. And one of the important things is endoplasmic reticulum is also equally important, like cytosol for synthesizing proteins, and secretory protein synthesis is the one which is made by endoplasmic reticulum. And whenever there is an excessive protein synthesis or improper covalent modifications, particularly once the proteins are made and if they're not properly folded because of lack of modification, or an improper degradation of the protein, or the changes in intracellular calcium, then what happens is there could be accumulation of unfolded proteins that promote the ER stress, and the ER stress could be acute or chronic, and they generate unfolded protein response. That could, depending on the stress, the cell may be surviving, or if it is unable to correct the protein folding defects, then it will lead to cell death. Having said that, we have actually reviewed a long time ago. The ER has 3 stress sensors. One is ATF6, another one is POC and EIF2 alpha kinase, and also another one is IRG1. To start with, they're all bound with BIP and ER chaperone, and they're inactive. But once the unfolded proteins are accumulated in the stressed ER, then the BIP, which is bound in ER chaperone, comes out of it and they get activated. And ATF6 actually promotes also the CHOP and some of the anti-apoptotic proteins. Also, POC phosphorylates. See, out of these 3 sensors, ATF6, IIU1, and POC, POC is the first one to get activated because its activation would result immediate shutdown of protein synthesis, the ongoing protein synthesis. So there is not much of a burden on the cell to fold the proteins. So that is what happens. And the other 2 are also equally important, and they also, upon activation, IIU1 produces actually XBP1, which is a transcription factor, which is much better put in the XBP1, and here. POC activation would result in ATF4. ATF6 on activation actually go to Golgi and gets cleaved, and the cleaved one becomes an activated transcription factor like ATF6P50. You see, this is a cleaved ATF6. And these 3 transcriptional factors, actually, they go to the nucleus and they facilitate the expression of UPR genes, which include chaperones, foldages, ER-associated degradative enzymes, amino acid metabolism, autophagic genes, apoptosis, and redox metabolism. So these are all the genes that are triggered in response to the ER stress. But what you see is actually is part of ISR, UPR has 3 stress sensors, but one of that is the ISR, where EIF2 is phosphorylated, ATF4 is made, or CHOP is made, or GAT34 is also made, which is actually acting like a phosphatase and stimulate the phosphorylation. So what we were trying to do is we actually measured one time in chronological aging what happens to the translational initiative. And we find most of the initiation factors that are downregulated. These are from various tissues, cortex, cerebellum, lung, liver, kidney, heart, spleen, of suckling, young, adult, and old. So you see, whenever if you are aged or whenever in all the old rats, the EIF4D is coming down gradually, and also S6 is coming down, EIF2 alpha levels have come down, EIF2B epsilon levels are coming down, EIF5 levels are coming down. But the kinase levels were somewhat increasing if you compare in suckling and adult and old. And also the PKR levels is also the EIF2 alpha kinase that is increasing. So what is the net result? Because of the decline in protein synthesis, initiation factors, there may be a decline in protein synthesis, which is true with aging. Actually, the protein synthesis comes down. So one of the factors could be there is a decline in the initiation factors synthesis. And then we evaluated the most important phosphorylations, the phosphorylation of EIF4E, 4GPP1, and phosphorylation of S6 and phosphorylation of EIF1. And all of them, EIF4E phosphorylation comes down, 4GBP phosphorylation. Hello? Hello? Okay. Hello, am I audible or not? Hello?
Yes, sir. Yes, sir. You are
Yes,
audible.
sir, you are audible.
So when we have analyzed actually the phosphorylation status of the initiation factors, particularly for E4E, BP1 and S6, you see, enhanced phosphorylation of them is actually a signal for increased protein synthesis, but they are coming down. But what we see actually when phospho-EIF2 alpha is also coming down. The decline of that actually suggests probably there could be a better protein synthesis. Maybe there is a better protein synthesis, but gradually it is unable to mount any more response, or it is not able to promote phosphorylation. That is what we try to investigate here. But what happens is the phosphorylation of EIF2 alpha comes down, and accordingly the levels of ATF4 are also coming down. And GAT34 levels are increasing. The phosphatase levels were increasing, actually with old tissue. You see, this is all old or adult and old compared to young. In most of the cases, in all the tissue, GAT34 is increasing the EIF2 phosphatase. And CHOP again is a death stimulating protein that is also increased. Phospho-JNK, which is death stimulating, is increased. BIP and ER chaperone, which is coming down. So what we suggested is these cells must be doing an active protein synthesis, but they're unable to fold their proteins right, and there is not enough chaperone level there, and UPR is not efficiently mounted. If it is efficiently mounted, then you will see EIF2 getting phosphorylated. Although the kinase levels are high, we also figured out the ability of these kinases to phosphorylate EIF2 alpha is coming down, probably because of factors like CHOP and also other regulatory proteins which might be inhibiting the phosphorylation of EIF2 alpha. So overall, what we suggested is the UPR decreases during aging. That is the cause for several metabolic and neurological disasters. And also the EIF2 phosphorylation is an adaptive signal. And see, persistent EIF2 phosphorylation can lead to cell death, but it is the reduced phosphorylation we have observed here, which is coinciding with increased pro-apoptotic proteins or cell death proteins. And then there was also another paper which came actually and by Han et al., who showed that what happens actually whenever there is phosphorylated EIF2, EIF2 alpha can be phosphorylated and phosphorylation blocks translation. And then the UPR is evoked, chaperones are made, anti-ROS products are made, and that leads to cell survival. But whenever EIF2 alpha phosphorylation is not there, then the cells will do a very good translation and there could be more proteins produced beyond the capacity of the folding. That is what happens actually in secretory protein biosynthesis, which are professional secretory cells. They produce more and more proteins and eventually they die much more quickly than other cells like neurons and others. And that because of the generation of loss and accumulation of unfolded proteins. So unphosphorylated EIF2 alpha is a death signal and phosphorylated EIF2 alpha is a survival signal. And then in the virus infection also, people have predicted that the virus always tries to replicate and the host wants to die once the virus infects. Accordingly, what you see, the viruses make several proteins. For example, adenovirus, HIV, Epstein-Barr virus, hepatitis delta virus, they make several RNA analogs and they can inhibit the PKR activation. Like the PKR is a double-stranded RNA-dependent EIF2 alpha kinase. It gets dimerized and phosphorylated, autophosphorylated, and this autophosphorylated one phosphorylates EIF2 alpha. But whenever some of these viruses make some proteins, like HIV1 TOR protein, Epstein-Barr 1 protein, HDV RNA, they can bind to the inactive one. And in the presence of double-stranded RNA, then PKR gets blocked. It cannot become phosphorylated. Similarly, the dimerization, some of the proteins, the viral proteins actually inhibit the dimerization of PKR. And some of them inhibit the autophosphorylation activity, like because they resemble like pseudosubstrate, vaccinia K3 and HIV TET. And there could be also inhibition of activation and sequestration of dsRNA. Some of the proteins, they actually sequester the double-stranded RNA so that PKR doesn't become active. Some of them are like phosphorylated to dephosphorylate. And that is how the viruses have devised several tricks to overcome the host cell AIF2 phosphorylation. Within that context, what we have observed was because we were expressing actually or using baculovirus expression system to express AIF2 alpha protein. And particularly one of the proteins, the phosphomimetic form, we could overexpress actually in insect cells, but not in mammalian cells. And that time we thought, how is that we are able to express the phosphomimetic form in insect cells, because this is toxic actually to the cells. Then at that time we were looking into the literature that baculovirus is a P35 protein, which is an anti-apoptotic protein that inhibits actually the caspase activity. So one of our colleagues had the P35 mutant virus, he spared for us, and then we tested the P35 mutant virus and wild-type baculovirus infection, and we found most of the stress-induced EIF2 phosphorylation is enhanced further by baculovirus mutant one, particularly P35 knockout virus infection stimulates stress-induced EIF2 phosphorylation in cell death, whereas the wild-type one protects it. It is likely EIF2 phosphorylation. Now we have not understood whether EIF2 phosphorylation is a cause or a consequence of caspase activation. And we have done several studies and we find ultimately actually it is both a cause. Initially, stresses can stimulate EIF2 phosphorylation. And that phosphorylation could be turned over by the phosphatase as far as is within the limits. But when there is a persistent stress sometimes, that leads to caspase activation. Caspases in addition can trigger again the kinases, because we have observed or people also have reported the kinase is also a substrate for caspase and cleaved kinase could phosphorylate much more efficiently the substrate sometimes. So that is how we have seen many of our observations actually support EIF2 phosphorylation could be a cause and a consequence of caspase activation. And we also find another macroviral protein, PK2. This also inhibits host cell EIF2 phosphorylation. And UV stress-induced phosphorylation is found reduced actually by wild-type PK2 virus, but not by mutant virus. And also the stress-induced cell death is reduced by PK2 wild-type virus, but not by mutant virus. So that is how we find actually the viral proteins. So they protect the insect cells. Here we use Sporoptera frugiparia, the insect cells, to study their survival and death. And I'm not going into the details of all this, but they all suggest actually there is a caspase activation to start with, and the caspase, when it is turned on, it will activate the EIF2 phosphorylation. So there are several stresses initially we studied that can affect the nucleus, ER, and cytosol. But we haven't tested what happens actually when mitochondria is stressed, whether an ISR pathway is the ISR pathway, basically the EIF2 phosphorylation, expression of transcriptional factors like ATF4, CHOP, and GAD32, and then translation and cell survival and death, which in turn stimulate. Of course, all these things can happen. So the ISR pathway, whether it is evoked in response to mitochondrial dysfunction, that was a question recently we asked, and we find actually the CCCP we used, one of the mitochondrial inhibitors of oxidative phosphorylation, and we find the EIF2 alpha is phosphorylated in response to CCC. And this is in response to tunicamycin. Actually, this is an ER stressor, tepsigargin. And in the absence of the tepsigargin, only CCCP is there, then you see. And tepsigargin is used as a control because we know tepsigargin as an ER stressor can also stimulate EIF2 phosphorylation. So that is what we see. The CCCP can stimulate EIF2 phosphorylation. And but then we were asking whether CCCP is stimulating EIF2 phosphorylation. Is it mediated by cytosolic kinases? Cytosolic kinases are HRI, GCN2, and PKR. Whereas the ER stress kinase is the PERK kinase. And also IIA1 is one of the things which get activated, which is an endonuclease actually, and activity it has. And that cleaves XBP1 mRNA. So we try to see if mitochondrial stress can evoke ER stress indirectly. So some kind of an interorganellar communications we were trying to assess also. And we find actually if you use tepsigargin ER stressor, then you can see tepsigargin within 8 hours it cleaves the XBP1 mRNA. Whereas in the presence of CCCP, you don't see that happening. So tepsigargin promotes ER stress, but not CCCP. So that is one. And also the PERK activation is measured here by its aggregation and mobility and the gel. And in the case of tepsigargin, PERK activation occurs because you see a higher mobile form here. I mean, which whose mobility is reduced actually. The PERK aggregated form. And in the case of CCCP, there is no such aggregation of PERK and no change in the mobility. So based on that, we said CCCP does not activate PERK. That is one thing we ruled out. Also based on XBP1 splicing. And then that is whatever EIF2 phosphorylation we are seeing is independent of PERK. And then we try to see if GCN2 is activated. We use GCN2 knockdown cells. And GCN2 knockdown cells, you see, UVB actually also can activate GCN2 kinase. That is what you see. EIF2 phosphorylation occurs in the wild type cells that have GCN2. whenever you give UVB irradiation, EIF2 is phosphorylated. But in the knockout maps, you don't see the EIF2 phosphorylation. And the CCCP presence also, we haven't seen much of an EIF2 phosphorylation here in the presence of GCN2, particularly the null cells where GCN2 is not there. We don't see much of an EIF2 phosphorylation increase. Whatever increase, what we have seen is the presence of KRB buffer, which actually mimics amino acid starvation, and GCN2 kinase gets activated and EIF2 alpha is phosphorylated. And if this phosphorylation, if you compare in the presence of CCCP in GCN2 null cells, it is comparatively less. So GCN2 kinase is also not involved in CCCP treated cells. And that is what we have concluded is not mediated by either PERK or GCN2. And then we look for HRA kinase activation. And interestingly, we find HRA kinase is activated, and here we use the HRA knockdown because the antibodies were not working very well. So we had to use HRA knockdown, and we find the HRA knockdown is good, and CCCP induced EIF2 phosphorylation could not be seen in the knockdown cells compared to the controls. Controls, you get a good phosphorylation, but in the knockdown, you don't see that. So we thought it must be HRA. And then we also ruled out the other possibility. Of course, we evaluated for other markers as well, like ATF4 and CHOP production, which is made in response to CCCP. They are all declined actually whenever HRA is not there. Or in HRA knockdown cells, CCCP cannot stimulate much of EIF2 phosphorylation and don't get much of EIF2 phosphorylation, and you don't express ATF4 in CHOP. So HRA, we thought that must be the kinase, but then we also looked at PKR activation, and the PKR recently is also shown like PERK. You can actually see its activation on the SDS space mobility, and in salt stress, it moves higher up and it is activated. But in the presence of CCCP, you don't see that kind of migration. And also the EIF2 phosphorylation levels are not stimulated. What you see in salt stress, it is stimulating, but in the presence of CCCP, you don't see much of the stimulation. So we have finally said, actually it is HRI, which is important. So, which is getting activated in response to cytosol. And it's a rare thing because, as I said, HRI is mainly limited to erythroid cells, and recent reports are coming HRI may be important in getting activated in some of the tumor cells only. And then we look for other signals as well. What happens in the presence of CCCP on EIF4 GBP phosphorylation is also downregulated in the presence of CCCP. AKT phosphorylation, which is also going to be through PI3K kinase pathway, that is increasing. Although there is an initial decline, but the AKT phosphorylation is stimulated in the presence of CCCP. And we also saw that the autophagy markers are increasing. CCCP usually have to phosphorylation, produce LC32 levels going high, and the protein synthesis is coming down when we measured the cellular protein synthesis using radioactive labeled material. So EIF2 phosphorylation correlates very well to the protein synthesis as well as EIF4 EBP1 phosphorylation, because 4BP1 phosphorylation, if it is high, that means protein synthesis is high, but it is low here. And the low levels also are showing up in terms of low protein synthesis. And the differential regulation, now we were looking at whether also AMPK phosphorylation, we looked at it because whenever there is a mitochondrial dysfunction, ATP levels can go down and AMPK phosphorylation can happen. That can also stimulate autophagy or cell survival. in addition to ADF4. So we wanted to test first whether AMPK phosphorylation is going up. Yes, indeed, in CCCP treated cells, the AMPK phosphorylation is going up. But Hepsigargan, if you take ER stress marker, where EIF2 phosphorylation occurs, dip expression takes place there, ER chaperones. Here, the AMPK phosphorylation is not happening. But here, in CCCP treated cells, AMPK phosphorylation is happening and ADF4 expression is happening just like in Hepsigargan cells. And the cells are actually showing up more of autophagy markers like LCP2 levels are high. And 4-EBP1 phosphorylation here is coming down, whereas in Hepsigargan treated cells, there is not much of 4-EBP coming down there. But in CCCP treated cells, the phosphorylation is coming down. So we wanted to really see what is that happening here. And we used actually two of the inhibitors. One is NAC, N-acetylcysteine, which is an antioxidant, where the CCCP is promoting any oxidative stress. And the other one is SRIB, a recently discovered ISR inhibitor. And this SRIB compound is really making this, in the sense it is found to enhance memory in rats and in mice by improving cognitive defects caused by shocking brain injury and trypan-induced neurodegeneration. It also reverses age-related memory decline. It blocks the ISR arm of UPR-induced expression of ADF4 in CHOP. It does not diminish EIF2 phosphorylation, but activates the EIF2B protein by promoting a decameric EIF2B complex from the subcomplexes, thus compensating for the inhibitory effect of EIF2 phosphorylation. This is what is known about ISRIB, is a memory enhancing compound, and it blocks ISR pathway by essentially stimulating the 2B activity, and it doesn't cut down on EIF2 phosphorylation. So we use these two inhibitors, and we find most of the time NAC completely reversed all the effects of CCCP, whether that be on EF2 phosphorylation, AMPK activation, AD2, EF4 expression, etc. The whole entire ISR pathway is mitigated, activation of that in the presence of NAC. CCCP induced ISR markers, all of them have come down. That is one thing. And then we looked at ISRIB. ISRIB, we saw differential effects now. ISRIB, what is that we find if you just go back, yeah. I should have put that together. Yeah, 4-EBP1 phosphorylation, for example, if you look into, it is partially restored. You know, this is in the presence of ISRIB we are talking about. And CCCP has completely cut down the 4-EBP phosphorylation, but in the presence of ISRIB, it is partially restored, whereas NAC completely restores it. That is one thing. And then the EIF2 phosphorylation, if you look at it, NAC completely inhibits EIF2 phosphorylation. But what we observe here is the EIF2 phosphorylation is still there. Although it started off at a slow rate, but eventually you get good amount of EIF2 phosphorylation. ISRIB does not inhibit EIF2 phosphorylation. What it inhibits is definitely the ADF4 activity it inhibits, or the ADF4 expression, CHOP expression. And so the ISR pathway is inhibited, essentially stimulating the 2B activity. That is what we could get, because EIF2 phosphorylation, however, is not shut down. And people have said that it is stimulating 2B activity. Accordingly, we see once the protein synthesis is restored, ATF4, the ISR expression genes are mitigated. The expression of ISR, ADF4 and CHOP are mitigated. And that is also shown by mRNA levels. And we try to understand how is that possible actually it is able to. slow down the rate of EIF2 phosphorylation, we find an explanation that in the presence of ISRIP appears to be due to an increased rate of EIF2 GDP recycling to EIF2 GTP by EIF2B. Okay, accordingly, we also found translation, but most of the important thing is the AMPK phosphorylation, which is cut down by NAC, but not by ISRIP. Suggesting that hello. Hello. Hello.
Yes, sir, you are audible.
Okay, I see background. Now, I'm essentially compiling the information here in a summary chart that what we have said was CCCP promotes the ISR pathway, which involves HRI activation, EIF2 phosphorylation, 2B activation, protein synthesis inhibition. And then it produces ATF4 and it leads to autophagy. And then autophagy, after some time, persistent EIF2 phosphorylation can lead to death. But this death is necrotic cell death. That is what we have observed. Not like apoptotic cell death, not caspase mediated. Of course, autophagy is because of LC32 levels decreasing, but autophagy leading to cell death is a kind of necrotic cell death that we have observed in the presence of CCP. In the presence of NAC and ISRIP, they are increasing. Actually, the cell death is reduced and the surviving cells are increased. But NAC mitigates all the effects of CCCP, whether that be AKT activation, AMPK activation, HRI activation, 4-EBP phosphorylation. They're all, kind of reversed by NAC. CCCP-induced effects are all reversed. Accordingly, the translation is restored. But ISRIP, when it comes to ISRIP, partially restores EIF4-EBP phosphorylation and slows down EIF2 phosphorylation. And AMPK activation is not at all restored. AKT activation is suppressed, definitely in the presence of ISRIP, suggesting that the autophagy that is produced in CCCP treatment, it is because of ATF4 expression, not because of AMPK activation. That is what we have summarized here. ISRIP, an activator of 2B, attenuates ISR markers, ATF4 in chalk, autophagy, slows down EIF2 phosphorylation, restores partially 4-EBP1 phosphorylation, and accordingly translation CCCP treated cells. Whereas NAC neutralizes all these signals, suggesting the involvement of ISR and CCCP induced autophagy. Initial decline in phospho-AKT and its attenuation by NAC and ISRIP. See, ISRIP doesn't have any reducing effect, but somehow we see some of the things are found like NAC, where it is able to reduce the phosphorylation of AKT initial stages and also able to restore EIF4-EBP1 phosphorylation. We suspect that it may be producing some amount of NADPH because of activating a pentose phosphate pathway or because of the proteome alteration in the presence of ISRIP. It may be generating some proteins that generates antioxidants. That is what we concluded. And this is a recent paper, and I wanted to acknowledge all my students, mentors, granting agencies, and UGC for the UGC BSR Faculty Fellowship post-superannuation. And I thank you for your patience. And all I wanted to say is this paper showing that CCCP-induced HRI activation occurs. We missed actually 2 papers have come recently side by side in Nature. I don't know if I have mentioned that, of course, in my abstract, extended abstract I mentioned. One is by Goa et al. And the one is by Cellular et al. I think these are the 2 papers which have shown that they also use mitochondrial dysfunctional agents like oligomycin, and they have shown HRI kinase can be activated in those cells. And there are 2 papers side by side in Nature. There, I think I wanted to end the talk. If you have any questions, please. Hello.
Thank you, sir, for such an informative and educative session. As we are running short on time, I would request all our honorable speakers to restrict their talk for 15 to 20 minutes. Now, moving forward, we have with us our next speaker, Professor Chandi Mandal from Department of Biochemistry, Central University of Rajasthan. Professor Mandal has done his master's in biochemistry from Calcutta University and obtained his PhD from IIT Kharagpur. Dr. Mandal's academic endeavor is not only reflected in the publication of his research work, he is also recipient of Investigator Award from American Society for Bone and Mineral Research. His research work has also been highlighted multiple times in newspaper and television. He is member of several professional societies such as Indian Association of Cancer Research, Biotech Research Society India. Dr. Mandal's research interest is aimed at understanding the cellular signal transduction mechanisms involved in breast cancer, bone metastasis, and bone remodeling. Now, I invite Professor Mandal to begin with his talk for the day.
Good afternoon. Can you hear me?
Good afternoon, sir. Yes, sir, you are audible.
And my slide is visible?
Yes, sir, slides are visible.
Thank you for giving this opportunity to share my research work with you. Before I start, I would like to give thanks to whole organizing committee, especially to Vice Chancellor, Dean Seth, HOD of Department of Biotechnology, and also my extended thanks to Professor B.C. Tripathi for inviting me. So today, just before I say that I am also an alumni of Sardar University. Actually, I joined Sardar University in 2012 September, and I was there almost for 1 year. Then I have moved to Central University of Rajasthan. So today I'll be talking on the title, Targeting Cholesterol Dysregulation Promises a Potential Therapeutics for Bone Metastasis of Breast Cancer. So my talk is related how the cholesterol dysregulation can be linked with the breast cancer bone metastasis. So you know that cancer is the biggest problem in all over the globe, including India. And in every month, we celebrate different type of cancer awareness program. In fact, in October months, we do celebrate breast cancer awareness as well as the liver cancer awareness. If you read very carefully, then every family, at least one member is either suffering from cancer or they are dying from cancer. So it's a huge problem for the economy. So before I start, I just would like to give a brief introduction that how the cancer can be caused or what are the factors for causing the cancer. So you can see over here that the environmental factors like pollution, toxin exposure, meat consumption, alcohol consumption, physical inactivity, smoking, these are the factors can be positively linked with the cancer incidence as well as cancer death. On the other hand, some biological factors or intrinsic factors like over body weight, obesity, cholesterol, diabetes, as well as the family history, that can also aggravate the cancer incidence, cancer growth, metastasis. So today I'll be talking the one of the intrinsic factor, that is cholesterol, how it can be linked with cancer growth as well as the bone metastasis. So when I start my research at Central University of Rajasthan at that time, I told to my students, can you do a map of top 50 countries which have highest cancer mortality rate? And he has done the map. And we have noticed that these red colors country are the top country which have highest cancer mortality rate. And we have noticed that the majority of the country, they are located from topic of cancer towards the North Pole or topic of cancer towards the South Pole. Then similarly, when we map the least 50 countries which have lowest cancer mortality rate, they are located in between topic of cancer and topic of Capricorn. So their locations are completely different. One of them are located from towards the North Pole or South Pole, and the least 50 countries, they are located towards the equatorial zone. So then similarly, when we map the top 50 countries which have highest average cholesterol in their blood serum, then we have found that the top 50 countries which have highest average serum cholesterol, they are located from the topic of cancer towards the North Pole. It is almost similar to the top 50 countries having highest cancer mortality rate. Similarly, the least 50 countries having lowest average cholesterol, they are located in between topic of cancer and topic of Capricorn. So they are almost similar to the least 50 countries which have lowest cancer mortality rate. So from this geographical location, we may say that there could be a link between cancer and cholesterol. Then when we have searched the literature, we had noticed that the high cholesterol rich diet can show the increase of cancer of various types like pancreatic cancer, colon cancer, rectum cancer, lung cancer. Similarly, elevated serum cholesterol, LDL cholesterol, may also increase the risk of several cancer, including breast cancer, testicular cancer. Recent studies also suggested that even cancer cells watch their cholesterol level. So we have done different kind of statistical analysis, worldwide data, and we have noticed that average total cholesterol and cancer mortality rate, they are positively correlated when we analyze by univariate analysis. It does indicate that as the cholesterol increases, the cancer mortality rate increases. Similarly, we have measured the similar type of correlation coefficient in case of site-specific cancer like lung cancer, bladder cancer, ovarian cancer. We had noticed that the positive correlation exists between cholesterol and different site-specific cancer. Next, we want to know what could be the mechanism. So we had noticed that the cancer blood sample, they have highest cholesterol level as compared to the normal healthy blood sample. This is the data we got it from the cancer patient sample. And similarly, the breast cancer tissue, they have highest cholesterol level as compared to the benign breast cancer tissue. So it does indicate that the cholesterol level is linked with the cancer malignancy. Similarly, when we have compared the cholesterol level between low metastatic breast cancer MCSF7 cells and high metastatic breast cancer MDMB231 cells, we have noticed that high metastatic breast cancer cells showed elevated cellular cholesterol as compared to the low metastatic breast cancer cells. So this data suggested that the cholesterol level is not only linked with the malignancy, it can be linked with the metastasis also. Then we wanted to know how the cholesterol are more in the cancer cells, what could be the mechanism. Then we have focused our attention on different cholesterol regulatory gene molecule, like the HMG CoE reductase. It is the rate limiting enzyme in the cholesterol biosynthesis pathway. LDLR, this is the low density lipoprotein receptor, which is involved for the internalization of the cholesterol in different cells, including cancer cells. And one transcription factor, steroid regulatory binding factor 1. So when we compare this gene expression in malignant breast cancer and benign breast cancer, we have noticed that malignant breast cancer tissue have elevated level of HMG CoE reductase, LDLR, and SRPF1. Similarly, we have noticed that The metastatic breast cancer has highest level of HMG-CoA reductase, LDLR, and SRBF1 as compared to the low metastatic breast cancer cell. So all these data suggested that the cholesterol upregulation is due to the dysregulation of cholesterol regulatory gene in the cancer tissue, and it is more dysregulated in malignant or metastatic cancer cells. Then the question whether the lowering of cholesterol can prevent the cancer growth. So for that, actually, we have used one cholesterol lowering drug, that is statin, especially simvastatin. The simvastatin inhibits the HMG-CoA reductase activity, and we have used nude mice. And in this, it's a immunocompromised mice. In this mice, we have injected simvastatin 5 mg per kg body weight intraperitoneally once a day for 7 days. After 7 days, we have injected breast cancer MD-MB231 cells to mammary fat. After 3 weeks later, we have isolated the tumor and we measured the tumor, and we have noticed that simvastatin treated group showed significant decrease of tumor volume in right breast as well as the left breast. So these data suggested that the statin, especially cholesterol lowering simvastatin, prevent the breast tumor growth in mouse model. To know the mechanism, we focused our attention on PI3 kinase AKT signaling pathway, since the PI3 kinase AKT signaling pathway is overactive in cancer cells. And we have noticed that the tumor suppressor P10 is upregulated in the tumor sample of simvastatin treated mice as compared to the tumor sample of control mice. Similarly, the PI3 kinase AKT level, especially the phospho-AKT level, was downregulated in simvastatin treated mice as compared to the control mice. And we have also measured the NF-kappa B activity. Finally, we had concluded that simvastatin upregulates tumor suppressor protein P10 to shut down PI3 kinase AKT pathway, which eventually inhibits the NF-kappa B activity. As a result, the breast cancer growth is inhibited in response to simvastatin treatment. So this is the observation we had published earlier. Since you know that 85% of the breast cancer patients die mainly due to bone metastasis, they don't die due to the primary tumor. So that is why we have asked whether the simvastatin can prevent the breast cancer bone metastasis. So to do this, we have again done the mice experiment. And in this case, we have injected again simvastatin 5 mg per kg body weight once a day for 7 days. Then we have injected the mammary MD-MB231 breast cancer cells in the left cardiac ventricle so that breast cancer cells can go to the blood circulation. After 4 weeks later, we have performed the radiological detection by X-ray using Faxitron machine, and we have measured the bone metastasis by Faxitron machine, and these black spots indicates the bone metastasis. So you can see that the control that this is the leg of the mice. So you can see that the control group where the breast cancer cells were injected, that they have shown the osteolytic lesions in the hind limb of the control group of mice. But in case of simvastatin treated mice, we have not seen this black spot, means simvastatin treatment significantly blocked the breast cancer bone metastasis. In fact, it was the first observation we have published in Journal of Biological Chemistry that simvastatin prevents breast cancer bone metastasis in mouse model. So how the bone actually remodeling happens, you know, the bone, there are 2 types of cells, especially the osteoblast cells and osteoclast cells. Osteoblast cells form the bone and osteoclast cells resolve the bone. In fact, osteoblast cells also secretes 2 cytokines, that is CSF1 and rank ligand. These cytokines bind to the receptor present in the osteoclast cells. Those are CSF receptors or rank. These ligand receptor interaction activate the osteoclast activity, and that osteoclast activity degrade the bone. So there is a balance between bone formation and bone decay. But in bone metastatic condition, the breast cancer cells can come to the bone microenvironment, and breast cancer cells can release many cytokines. One of the important cytokines is CSF. Their CSF can binds to osteoclast cells and can aggravate the osteoclast activity. And when the osteoclast activity is more, then bone will be degraded, and the degraded material and degraded factors can bind to the breast cancer cells, and it can aggravate the breast cancer potential. So there will be a vicious cycles. So if you want to block the bone metastasis, so we have to block the vicious cycles. Thus, we thought whether our simvastatin can block these vicious cycles to prevent the breast cancer-induced bone metastasis. So to do this thing, we have done the experiment using osteoclast precursor raw 264.7 cells, and we have added breast cancer condition medium, and we have noticed that breast cancer condition medium can increase the osteoclast activity. This is measured by trap staining. The trap activity indicates the osteoclast activity. It's a multinucleated osteoclast. And there are many enzymes, those are involved in osteoclast activity. One of them is TAP, another is the cathepsin K. We have noticed that the breast cancer condition medium can increase the TAP transcript level as well as the cathepsin K level. And one of the crucial transcription factor, it is known as osteoclast transcription factor, that is NFAT-C1. And NFAT-C1 actually regulates the osteoclast activity. And we have noticed that breast cancer condition medium can increase the NFAT-C1 transcript level. Now the question whether simvastatin can prevent the breast cancer-induced osteoclast activity. So we have done again the TAP assay and TAP staining. We have noticed that condition medium can increase the TAP activity, whereas the simvastatin treatment can increase the breast cancer condition medium TAP activity. We have also used another drug, that is MBCD, that is cholesterol depleting drug, and that MBCD also block the breast cancer-induced TAP activity. To know the mechanism, we have again focused our attention on different molecules like TAP, cathepsin K, and NFAT-C1. We have noticed that breast cancer-induced TAP activity was decreased by the treatment of simvastatin as well as by treatment of MBCD. To confirm, we have focused our two crucial enzymes, that is matrix metalloprotease 2 and matrix metalloprotease 9, which actually degrade the bone. And we have noticed that condition medium can increase the MMP2 level. It is done by Western blot and MMP2, and it is done by zymography. And but the simvastatin treatment or MBCD treatment can block the MMP2 activity in raw cells, that is the osteoclast precursor cells. So later on, we have noticed that the cholesterol depleting can also prevent the breast cancer cell survival, as well as it can also inhibit the apoptotic gene BCL2, BCLXL, but increase the apoptotic gene caspase-3 and BAX level. MBCD can also inhibit the cell migration. So the cholesterol depletion can prevent the cell migration, as well as it can induce the apoptosis. It can prevent the cell proliferation. We have also found that MBCD can inhibit the epithelial to mesenchymal transition. It is a very crucial for bone metastasis. It is crucial for metastasis. And we have noticed that MBCD can increase the epithelial marker E-cadherin and keratin 19 level, whereas it can decrease the EMT marker vimentin and E-cadherin level. We have confirmed that MBCD inhibits the stemness as well as the sphere formation by doing colony formation and sphere formation assay.
Pathogenic cultures.
So we have mentioned that the CSF1 is very crucial for bone metastasis. So we have compared the CSF1 level in ELISA experiment. So we have taken normal breast epithelial cell, MCSF10A cells, and low metastatic MCSF7 cells, and high metastatic MDMB23 cells. We have noticed that MDMB23 and high metastatic breast cancer cells has elevated CSF1 as compared to the low metastatic MCSF7 cells as well as the MCSF10A cells. Moreover, the bone-specific MDMB23 cells has more CSF1 secretion as compared to the parental MDMB23 cells. So this data suggested that CSF1 is not only related to metastatic activity, CSF1 is also related to bone metastatic activity. So that is why we have focused whether the simvastatin can target the crucial cytokine CSF1 in breast cancer cells. So we have noticed that simvastatin as well as the cholesterol depleting MBCD inhibits the CSF1 level in low metastatic breast cancer as well as the high metastatic breast cancer cells. To know the mechanism how the simvastatin actually regulate the CSF1 level in breast cancer cells, we focus our attention on NFAT-C1. It's a very crucial transcription factor. It's also increase the EMT as well as the cell migration. And we have noticed that the simvastatin treatment inhibits the NFAT-C1 in breast cancer cells. Similarly, the cholesterol depleting is also inhibits the NFAT-C1 level. And also one of the molecule that is the DKK1, it's the Wnt signaling inhibitor. There is a link between NFAT-C1 and Wnt signaling. And we have noticed that the simvastatin inhibits the DKK1 level in protein level. And when we have treated the cells with recombinant DKK1, we have noticed that CSF1 as well as the crucial other osteoclastogenic cytokine rank level is more in the breast cancer cells. And when we have transfected the constitutive active NFAT-C1 in breast cancer cells, we have noticed that constitutive active NFAT-C1 increase the DKK1 level, and it also increased the CSF1 and rank ligand transcript level. As a result, we have concluded that simvastatin or MBCD downregulate NFAT-C1 to downregulate DKK1, which eventually downregulate the CSF1. So this could be one of the mechanisms how the simvastatin inhibits the CSF1 expression in breast cancer cells. Similarly, when we have treated the cells with constitutive active NFAT-C1 transfected conditioned medium, we have noticed that the conditioned medium of constitutive active NFAT-C1 increases the TAP activity, whereas the cyclosporine A, it is a NFAT-C inhibitor which inhibits the TAP activity in raw cells, whereas the recombinant DKK1 can increase the TAP activity in the raw cells. So we have also confirmed by performing different kind of artificial experiment, and we have noticed that conditioned medium of constitutive NFAT-C1 can increase the TAP activity by upregulating TAP, cathepsin, and NFAT-C1 expression level in osteoclast precursor cells. To know whether this concept is true, and we have analyzed the tumor sample in breast cancer tumor sample, and we have found that the malignant breast cancer tissue has more amount of NFAT-C1, DKK1, and CSF rank ligand as compared to the benign breast cancer tissue. So based on this observation, we have noticed that the breast cancer cells actually upregulate NFAT-C1, which actually eventually upregulate the DKK1. This DKK1 upregulates CSF1 rank ligand. These two crucial cytokines binds to osteoclast precursor cells, and this binding activate NFAT-C1, which upregulate TAP. Eventually, TAP activity can upregulate cathepsin K and MMP, and this cathepsin K and MMP is responsible for osteoclast activity. And whereas the simvastatin inhibits the NFAT-C1, or cholesterol depleting inhibits the NFAT-C1, which eventually inhibits the breast cancer-induced bone metastasis or osteolytic activity. Before I ending, I would like to acknowledge my collaborator, like Professor Bizugulov from Russia, Professor James Radosevich, Professor Alonag, and Professor Gopal Kundu, and Dr. Mohavir Panwar. So before I ending, I would like to thank my students, my past students, my current students. So Ankit is my first PhD student. Nakosik is doing PhD now in Thailand. Tomo is doing postdoc in America, and these are the current PhD students: Sitoma, Sneha, Pooja, Seta, and Manas. So now I'd like to end my talk. If you have any query, question, please let me know. Thank you.
Thank you so much, sir, for your wonderful lecture. Now we have our third speaker with us, Professor Nilima Mondal. Professor Nilima Mondal has completed her PhD from School of Life Sciences, Jawaharlal Nehru University, New Delhi in 1999. Her area of interest includes cell biology and cancer biology. She is currently working as an associate professor in Department of School of Life Sciences, JNU. She has received Assistant Professor Award in the Department of Biomedical Informatics from Ohio State University, Columbus, USA in the year 2011 and 2013. She has recently published a reviewed article on a unique 45 amino acid region in the toprim domain of Plasmodium falciparum in the year 2009, and a reviewed journal on designing novel inhibitors against histones acetyltransferase in the year 2017. Now, I would like to welcome ma'am to address the audience.
Hello, everybody. Can you hear me?
Yes, ma'am.
Okay, and I have shared my slides. Are you able to see it?
Yes, ma'am.
Okay, let me start. Are the slides one sec. Are the slides visible?
Yes,
Hello?
ma'am. Yes,
okay. So,
ma'am, I request you, excuse me.
Yeah.
Ma'am, I request you to kindly zoom out your slides.
Zoom out.
Yes, ma'am.
1 minute. Is it? I just made a slide presentation. Is it okay now?
No, ma'am, it is not.
So let me just do this way. Is it visible?
Yes, ma'am, it is fine now.
This is fine?
Yes,
So
ma'am.
let me not go into the slide mode then.
Yes.
Okay. So basically, I'm going to talk about how we can alter the cell proliferation, and because it is one of the hallmark of the cancer cells. And I will try to be as short this thing and to the point as possible, because all of you are really hungry, right? So is it clear? Are you able to see the slides?
Yes,
Hello?
ma'am. Yes, ma'am.
Okay, okay.
Slides are visible.
So basically in cancer, we know that there is this deregulation of the cells either by genetic changes or epigenetic changes, because of which there is an uncontrolled cell proliferation, and that leads to, in an extreme case, it leads to cancer. Now, so this thing tells us that if I can block the cell proliferation in some way, I will be able to control or combat the cancer. So to work in this direction, what we have done, we have, in collaboration with other scientists, we have used different compounds or synthetic compounds, peptides, etc., to block the cell proliferation and see whether we can control the cell cycle. So this is one of the examples where we have worked on the prostate cancer cell with this compound SH1. And we found that first what we do is we check whether the cells are affected or not, the cell viability is affected or not. So as you can see in this figure C, the cells were affected at 50% of the cells were viable at around a concentration of 60 or 70. Here we have used two cancer cell lines, prostate cancer cell line, DU145 and PC3. Next, what we did is we checked how the cells are affected, cell cycle is affected. Just this is for the students. The way we see the cell cycle is we do the flow cytometry, and on the y-axis we have the number of cells, and on the x-axis we have the DNA content, and based on that we differentiate the cells in different phases. And as this figure is showing, when in the control you see the cells, the sub-zero is very low. Hello? Yeah.
Yes, ma'am. Could you please switch to the slides, because it is still in the first slide page. You just switch.
Oh, is it now?
Yes, ma'am. Now it is the slides.
Now slides are showing?
Slides are showing, but they are zoomed in, so that only the.
I haven't zoomed in.
Ma'am, you can directly go on to the another slide from the PowerPoint by clicking the slide number 3rd page or the 4th page.
That's what I'm doing. Is it now moving slides? Are the slides moving? Hello?
No, ma'am, slides are not moving. Ma'am, if you will select it from the corner.
Let me close down this. and see if I can again go back to that. Now I'm not able to do anything. Why this is so let me share it again. No, this is not the slide. I lost the slide somewhere in between. All is the slide visible now?
Yes,
Hello?
we are seeing the
Is
first.
the slide moving?
Yes, ma'am. The slides are moving.
Okay.
Thank you, ma'am.
So should I go this way or should I put in the slide mode?
Ma'am, proceed this way. It is much easier to.
Okay.
Thank you,
So, okay. So now I am in the 5th slide. Has the slide changed?
Yes, ma'am. The slides are changed. Cell cycle profiling.
Okay. Thank you. Sorry for this, but technical problems we all are facing with this webinar and all, right? But anyway, so now with this, this synthetic compound, when it was given to the cells, as you can see, this tetraploidy has increased a lot, and with the increase in time by 72 hours, most of the cells are dead. So at the IC50 value, by 72 hours, the cells are dying. Now, the problem with the cancer drugs are that we have a lot of side effect and systemic effect, because of which the patients are adversely affected. So what we did is we took a suboptimal subcytotoxic concentration of the compound, and so that is almost 6 to 7 times lower than the IC50 value, that is 10 micromolar, and tried to see how they are affecting the cells. And as you can see, by 72 hours, we have a lot of accumulations of cells in the S phase, right? As compared to the control where the DMSO treated cells. DMSO, why? Because it is the vehicle in which we have dissolved the compound. And this is the panel where these are non-cancerous cell lines. And as you can see at this concentration, these cells are not affected. Now, further, we wanted to see when the cells are arrested at S phase, how the cell cyclins are, cyclin and cyclin CDKs are affected. So as you can see here, the S phase cell cycle, the cyclin E and cyclin A are involved. So we checked about it, and so can you see the bands, please? Hello?
Yes, ma'am, we are able to see.
Okay, so cyclin A, as you can see, with increase in time, it is decreasing, but cyclin E is not affected. So cyclin A is affected. Even when we did the cyclin CDK, checked the kinase activity, we found that yes, cyclin A kinase activity with the increase in time is going down. Now we wanted to see the localization of the same. The cyclin A and CDK, which are involved in this, are affected when the compound is, the cells are treated with the compound. So you can see in the first panel, the DMSO treated cells, the cyclin A is totally localized in the nucleus, as well as cyclin D is there in the nucleus and a little bit in the cytoplasm. Now, when the cells are treated with SH1, what is happening is, you can see that cyclin A is really, really downregulated, and CDK2 has gone out in the cytoplasm, and it is also downregulated. Same is the case in the PC3 cell line also. So, and the merge, this thing panel also tells us that there is a crosstalk between CDK2 and cyclin A when it is just control in the control panel, but in the treated sample, it is not able to do its function. And that's why we were not able to see this kinase activity. So that means that SH1 is blocking the cyclin CDK activity, but how it is doing it? So next, we know that P21 is a protein which blocks the cyclin CDK complexes. Just cyclin D CDK complex is not blocked, that is induced, but cyclin E, cyclin A, and cyclin B1 are inhibited by the P21. So we wanted to see what is happening to the P21. And as you can see, yes, with the increase in time, the P21 is induced more and more. Next, we wanted to see that P21, we know that it is regulated by P53 and retinoblastoma proteins. Now, the beauty about this DU145 and PC3 cell line is that PC3 is P53 null. That means there is no P53 protein in this cell line, whereas in DU145, P53 is there, but no retinoblastoma cells. So what we did is we checked what is happening to these two proteins, which might be regulating the P21. And yes, what we found that in DU145, P53 is phosphorylated, which in turn might be inducing the P21. And in case of PC3, the phosphorylation of retinoblastoma is inhibited, and that's why the cells are not able to move, or this P21 is upregulated. Now, upstream of this P21 is BRCA1. So we wanted to see how the BRCA1 is affected. So as you can see, by 72 hours, P21 is upregulated, so is BRCA1 in both the cell lines. So we wanted to see next how these are localized whether it is localized in the cytoplasm or in the nucleus. In the control panel, the level of P21 and BRCA1 is very low. Now, when we are treating the cells with the compound, what we are able to see that there is a huge induction of the proteins. The BRCA1 has increased, very nice punct figure we can able to see here, fluorescent figures are there, as well as the P21 has been induced. And in the merge, you can see here very nicely, these two are merging, so crosstalk is going on. So that means that by here we say that BRCA1 is inducing P21, that in turn is blocking the cyclin CDK complex and blocking the cells at S phase. Now, if the cells are arrested at the S phase, we know that PCNA, the proliferating cell nuclear antigen, is a protein which is an essential or integral part of the DNA replication complex, right? It works as a clamp. Now, what is happening to that protein? If I say that S phase is arrested, that means that proliferation cell replication is not taking place. So it tells me that PCNA must be going down. And that is what we found, that here PCNA is by 72 hours, it is really low in both the cell lines. Now, this P21 here around this position, the P21 interacts with the PCNA, okay? So we wanted to see by pull down assay whether these two are able to interact when we are treating the cells or not. So we did the pull down with P21 and Western blotted with PCNA and P21, and we can find that with the increase in time of the treatment, the PCNA is not able to interact with the P21 anymore, right? Same is the case here. We can see in the nuclear localization or fluorescent data also, that when the cells are not treated, there is very nice expression of PCNA is there, and P21 is hardly we can see P21. But when the cells are treated with the compound, what is happening is P21 is upregulated. You can see that there is a nuclear localization of the P21, but PCNA is really going down. same is in both the cell line, the same kind of data we got, okay? Now, so up to here, what we saw that SH1, BRCA1 upregulated, that in turn is inducing P21. P21 on one hand is blocking the cyclin CDK complex and arresting the cells at S phase. At the same time, it is degrading this P21. The interaction is not there, because of which now replication blockage is also there. The replication is not able to take place. Now, we note that there are some stress kinases which are upregulated in case of when the cells are under stress, right? So what normally happens is one of those stress kinases, P38 MAP kinase. Now this P38 MAP kinase, when it is phosphorylated, that in turn phosphorylate HSP27, heat shock protein 27, and this heat shock protein 27, what it does, it blocks the apoptotic cells, apoptosis of the cells. And this is another way by which the cell number can be increased in the cancer cells. So we thought, can this compound can block this or not? So what we did is, we treated the cells with the SH1 and then checked whether the P38, what is happening to the P38 phosphorylation. And as you can see, by 72 hours, the phosphorylation of P38 is really drastically reduced. Same is the case with the HSP27. With the increase in time, the phosphorylation of the HSP27 is going down. Same, we have confirmed it with the fluorescent microscopy. As you can see here, that HSP27 and P38 are very nicely expressed when the cells are not treated with the compound. And when the cells are treated with the compound, both the proteins really go down, and thus it is corroborating with our Western blot data. Now, in that, if this is happening, so we just said that, we just said that if HSP27 is going down, then it in turn can, what will happen? Now the blockage of the apoptosis is gone. So that means apoptosis should take place, right? I'm not going into detail of all this, just here what we have shown that ROS is also upregulated, right? ROS and mitotic membrane potential is dissipated. This in turn tells us that yes, maybe the cell killing which was happening in the first slide where you saw that the cells, lot of this sub-G0 cells are there. So maybe the killing is because of the apoptosis. So the apoptotic markers we checked and we found that yes, PARP is cleaved. Here you can see with increase in time, the PARP cleavage is there. Caspase is also cleaved. So from pro-caspase now active caspase are formed. Then this survivin is also going down. BCL2 has also gone down in both the cell line. So that tells us that yes, the cell killing which is taking place by the SH1 is through the apoptosis. And at the same time, we checked that what is happening to the non-cancerous cell line. And as you can see, the PARP is not at all degraded here. That tells us that, okay, the non-cancerous cells are not affected. So far, this is in a nutshell what we have talked about, that SH1 affecting the P38, this thing, and this HSP27 and blocking the cell proliferation. The cell proliferation block is now removed. So the cells are dying now. And in this case also, the PCNA is degraded, so cell cycle arrest is taking place, and all these are converging to the apoptosis. Just here, other pathways also we have deciphered. I'm not going to talk on that, but basically what this compound is doing is it is affecting the cells in multiple pathways, but they are converging where the cells are dying because of apoptosis. Now, next, just in another 5-7 minutes, I will just give you a glimpse of same kind of work, but in different cell lines. Now here we have worked on this breast cancer with another compound, and we found again that there is a huge S phase increase. And here in this case, cyclin E is increasing and cyclin A is decreased. Okay. And in this case, again, this HSP27 phosphorylation is blocked and the cell death is taking place. Now, if that is happening, we checked it in vivo also, what is happening to the tumor growth. And as you can see, this blue line where the cells are not treated, where the mouse is not treated with the compound, and we can see that there is continuous increase in the tumor. When the mouse was treated with 10 mg per kg body weight of the compound, there is a slight reduction in the tumor growth. But when it was treated with 25 mg per kg body weight, the mouse, there a huge decrease in the tumor growth was observed. So this, and as we know that we checked also here, I have not shown the data, we cytotoxic effects have also we checked, and the mouse was not affected cytotoxic. There was no cytotoxicity because of this compound. So maybe further work has to be done to take it to the bedside. Now, in breast cancer, what happens is all of us know that tamoxifen is a compound which is, or a drug which is always given to the, most of the time is given to the breast cancer patients. And we know that initially the breast cancer patient very nicely respond to it because this tamoxifen mimics the estrogen and binds to the estrogen receptor, and now it blocks the cell proliferation. But what happens is with time, this cancer patient develops resistance towards this drug. And also it has been shown that the cancer patient develop endometrium cancer. What must be happening there is that this estrogen, this tamoxifen binding to the estrogen receptor, and in the uterine cells, there are other co-activators which are now binding to this complex, and the transcription or is taking place, because of which now the cell is able to replicate. Okay, and is able to replicate, because of which this endometrium cancer is developed. So we wanted to see, can we, by using this compound, or how can we overcome this resistance towards the tamoxifen? Here I have not shown about the data with the SH3 compound, but again, here before going to the data, what we observed that in case of tamoxifen resistant cell line, the P38 phosphorylation is really goes up. HSP27 phosphorylation also goes up. And this again gives us the clue that, okay, because of which now the apoptosis have been totally blocked, and that's why cell proliferation are taking place. So can we block it? So here, with the help of computational colleagues, what we did is they designed some peptides, and we wanted to check whether this is able to block this pathway or not. And to our pleasant surprise, what we found that this tetrapeptide FWCS, what it is doing is, as you can see, it is blocking the P38 phosphorylation. It in turn is blocking the HSP27 phosphorylation, and in turn there is this apoptosis which is taking place as shown by the degradation of this or cleavage of the full length path. Okay, so this is the pathway which is occurring, and so this tells us that yes, we will be able to overcome if we can use this peptide. Further work has to be done in this direction. Now, this is just last slide where I have shown that we are working with the gastric cancer also. And here, this H. pylori is a one which is continuous or infection of the H. pylori can cause the gastric cancer. Yesterday, we lost one of our scientists to the gastric cancer. But anyway, so what we have done, we have deciphered the pathway, and we have shown that this gastric cancer cell line, say for example, AGS, where people have shown that this c-MYC is upregulated and is beta-catenin and c-MYC are responsible for continuous cell proliferation. So we checked whether we are able to block it or not. And as you can see here, yes, we are able to block it very nicely. The c-MYC is totally down, beta-catenin is down. So, and we saw that yes, cell proliferation is also going down here with the increase in the concentration of the compound. Next, also we checked that in the cell migration also goes down, and I don't know whether you are able to see here or not. Very nice focal adhesion kinases are formed here when the cells are treated with the compound. What is happening here is the cells are able to form the focal adhesion kinases, but not able to dephosphorylate. Okay, so that they can take out, like detach from the place and then again go and attach, and like this it can move and form. So they are not able to migrate. And again, this compound is able to block the cell cycle. And as these markers are showing us, the PARP is again cleaved and the survivin is also going down, tells us that yes, apoptosis is taking place. So by use of different peptides and compounds, we can overcome the cancer's proliferation. But the only problem is that the side effect. And as we have shown that if we are using the suboptimum conditions, sometimes it can help us. So by this, I'm ending my talk, and just I showed that how we can alter the cell cycle and can control the cell proliferation. Before ending, I would like to thank my collaborators, Professor Amir Azam from Jamia Millia Islamia, Dr. Subbarao Naidu from JNU, and Dr. Preetham also from JNU. They have given me the compounds and the peptides and helped me in designing the compounds also. And here some of the work has been shown which has been done by these students and the funding agencies. I'm thankful to the funding agencies, DBT, UGC, and DST. And of course, I'm thankful to JNU Life Sciences Central Instrument Facility and AIRF Facility. Thank you, everybody, for your precious time. Questions, please.
Participants, do you have any questions? There are no questions, ma'am. Thank you so much.
Questions. Okay, everybody is hungry, right? Okay, welcome. Thank you very much.
Thank you, ma'am, for your enlightening lecture. Now we have our fourth speaker with us, Honorable Professor Dr. Pradeep Verma. Professor Dr. Pradeep Verma has completed his PhD from Sardar Patel University, Gujarat in microbiology in 2002. His area of interest includes microbiology. He is currently working as a professor in the Department of Microbiology, Central University of Rajasthan. He has also processed the production of high yield of biobutanol as European patent in the year 2012. He has recently published reviewed article on a comparative assessment of autoclave and microwave assisted peroxymetal complex in the year 2016. Now, I would like to welcome to address the audience.
Thank you very much for inviting me for this conference. I would like to thank the organizer, especially the Vice Chancellor, Chada University, and Professor B.C. Tiwari, who has given me a call regarding the talk to this particular conference. So regarding this, the overall, I would like to talk on this particular thing, that we are working on a biofuel especially, and there are a lot of factors in a biofuel. So one of the enzyme, the pre-treatment, and other part of is the application part. So our work is focusly in the pre-treatment and the enzyme, which is going to work on a particular aspect. So in this case, we are working on the several organism, especially those can break down the cellulose, hemicellulose, that can be converted into sugar and then can be evaluated product component like the prebiotics. So we have a very big project from the DBT, which is we have around 70 to 80 lakhs rupees. So we have isolated so many coordinated projects from Assam. So we have isolated the fungi from Assam region, which is especially origin, and we will want to see the proteome analysis of this particular fungi because it's a good producer of gyanases. So that's the overall summary of the particular work. So now I would like to go in a brief about summary of the work. So why we are looking for the biomass? Because as you know nowadays, because the gasoline price is regularly increasing, and day by day now, at the moment I can say that the biofuel which can be compatible to the current prices. So the objective was if it is possible, and the government also has relaxed the regulation. Now you can see that the 15% blended mode is available to all the petrol pumps. So now we had to increase the production part of a biofuel. So how the biofuel is produced? So depend upon it is the biomasses, which is the agriculture waste, which can consist of the lignin, hemicellulose, cellulose. And if you look into the complexity of the biomasses, we have to design our process. Number one. Number two is that the biomasses, which is consisting of 40-50%, which is cellulose, and 35 to 30% is around this hemicellulose and lignin, which is all the three components is useful. If you utilize all the component, then we can have a substantial progress in we can make in biofuel development. So what are the organisms which can be responsible for this particular biofuel? The bacteria or fungi. Most of them bacteria, bacillus species, and fungi, which is Aspergillus trichoderma species, which are learned for the Jalen product. So in our case, in this study, we have a focus especially the Aspergillus oryzae, LC1, which is we have isolated from Assam region, and we want to protein analysis for this and how it works. So that particular commercial gyran can be broken down by by hydroxy beta for 1,1,4 linkage, and they can be converted into the monosaccharide, oligosaccharide, yellow So then after the monosaccharide oligosaccharide can be converted into the by using yeast and the bacteria to the biofuel. So now if you look about this, the complexity of the biomass and how to achieve the task. So we have a pre-treatment developed by us, which is patented and commercially available in the market. So now we are also working on how to reduce further the cost. So we have to look, because 25 to 30% cost of the enzyme is responsible for that. So that is why our next target to reduce the cost of the enzyme. So if we can produce the in-house enzyme, so then we cannot depend on the commercial enzyme, which is very costly and effective, and which can make the product cost very high. So if you look about gynanes have a lot of uses. Paper pulp industries, you can say that textile industry, food processing, and animal feeds, digestibility, and other processes also they have been used. So this is the scenario. If you focus on a particular these two enzymes, cellulose, gynanes, and other enzymes also, which can be part of a breakdown of this complex material by plant biomasses, then it can be wonderful to achieve the task which we are looking for. So here we can see the background of why we think about that. So if enzyme global market is 6.3 billion 2017, so that is the target year was this. And now you can see biofuel 52.57%, which is the compound annual growth rate of a market is so high. So that is why we have to think about how to reduce the cost of the enzyme. So and you can see that we have annual the crop productions in there in the bar in 2024, you can see that how much pectic is produced on there, and it is the various state province of India which can take part in this, and the major is Uttar Pradesh, which is have a largest production of the sugarcane, all other biomasses are there, and rice straw, other components are there. So here, if you look about our objective was here rice straw, because rice straw is a staple food and they have been used in almost all part of India. So how to utilize the rice straw for the biofood purposes. So if you an estimated United States USDA approval, then 2017, that they have been around 1.5 kilogram rice straw per gram. Per kilogram rice grain harvesting. So you can see how much biomass is generated Number two. So global rice production, you can see around 481 billion tons. And the India contribute around the 22% this, which is a costing, which is around 106.16 million. So what we do in India, we generally do the burning after the harvesting the crops, and then we just start burning. And that is a critical pollution. Now, if we can alternately use this product and this waste for the enzyme production, and that subsequently to the bioethanol production, then it is a zero waste, utilizations. So that is why we had to focus on this. So now, what we have to use for the production of the xylanase. So we have been isolated the screening of the novel fungal strain for different unexplored area of Assam, which can be produce high amount of xylanase. Then for that we have to medium optimization we have done for the first one factor and multi CCD RSM. I will go in summarize here, and the enhancement of xylanase in the proteome, analysis of the novel enzyme xylanase strain, then identification of gene encoding xylanases, then recombinant DNA technology, cloning, expression of E. coli, and the characterization of recombinant xylanase. And then we can see how it can be used the application part of this purpose. So this is the summary of things. So that is the objective of the things, the selection of suitable media for production of xylanases, especially the isolated strain of Aspergillus oryzae, which was first to report by our group, and the optimization media and the nutritional effect, the one factor approach, the identification xylanase encoded gene, which is cloning expression by using expression coli, and the application of xylanase in certification, production the second generation biofuel, like ethanol production. So here we have been screen out the different media which has been used for fungal Aspergillus strain. So we have been used around 3 media, which is Tepedox, vessel salt media, media, and the composition reflected here, which has been done by a lot of PSPS worker, and the inoculation was done, and xylanase activity was measured using 1% xylanase substrate, and the reducing sugar was measured during DNS method, and protein also determined by the BSS as using as a standard. So here we can see the different component of M1, M2, M3 media component are there, and then we have factors are varied, and then we can see how does it work. And but only one time we have to change the factor at a time. So that is why we took other parameters to be constant. So here we have to see the result how we operate. So this is lentil plant where we have got this this fungi, which is Kachar district of Assam. Colony morphology, you can see that it's a greenish, and then you can see the xylanase, which is the colony when is there, utilize the substrate, then it's the purple color appears. So if you do the production, then you can see that in the 3 different production media, MSM, vessel salt medium, Tepedox medium. So we can see that the minimum salt medium, which is the best, which can give you the 225 unit per ml production of the 12th way. So that is the reason why we have work on the particular this media further. So down we have used this particular media. Now we have focused on one media only, and then we have used for one factor approach, minimum salt medium, and incubation 28 degrees for 12 days, because we have a maximum yield on 12 days, and this is RPM was the shaker bath. So we have 100 RPM over this. So we can see that the incubation time, these are the factors which you can see the ammonium sulfate concentration, here the potassium concentration, urea concentration, and calcium concentration, then magnesium. These are the parameters which we can see that they can be minimal and they can regulate the enzyme activity. So the overall, we can see that unoptimal media, if you look about the unoptimal media, we can find only the 225, which I already told you, the 225. Now after optimized media, we can see that around 5.7-fold increase the activity was there, and we see that as this phase electrophoresis and the nitrogen by we can see that there's a group of xylanase enzyme which can be working on this. Now we have to do the proteome analysis of the particular fungi, which is we do the LCMS studies, and then we can found a total 73 protein were identified by LCMS analysis. And functional classification, if you can see the functional classification, showed the hydrolytic enzyme consisted of the 48% is the glycosidic, and 11% proteases, and 1% polysaccharides. And other than the esterases, 9%, osteo, 30% other proteins are there. So you can see that the hydrolysis enzyme content, which is the major hydrolytic enzyme, hydrolysis, which is the major part of it. So now we have to look further about this. The total 26 family of glycoside hydrolysis, which is, we can see that, which is have containing of the serine, carboxylic esterase, peptidase, and oxidase. So these are the first time we have reported that this particular LC stain, which have a protein analysis like this, and which the study which has published, we can go in detail by in Annals of Microbiology, which is a highly cited paper of us. Then we are looking for the second objective, how that particular encoded gene can be cloned and expressed using histology. Then methodology was same. Then we isolate the nucleotide gene, and then we know isolation, identification of the encoded gene, primer designing. These are the common practice we do. I have to skip the details in this. So then we do the histology, DNA template, primer designing, and all these things, cloning morphology, and we have to overexpression purification, characterization of this, transform in host E. coli BL21D3. So these are the things which we have performed, and these are the peptide sequence obtained from the secretome analysis of aspergillus ITC, this LCM analysis. So these have been done by us. So then after that, we can sound that XY and F1 gene, aspergillus RB 40 used to designing primer, report assist number this. Then partial sequence of this 5.8 ribosomes genes, complete sequence ITS gene, and stain on this accession number this. The nucleotide and reduced amino acid sequence X1 gene underline the region, which can be, these are the underlying region where we can say LCS amino acid extra proteome native generation from the aspergillus RIG ITC 854. We have deposited this culture because in a culture collection, because it's a very unique culture which is producing high amount, and which is the stain number has been allotted to us. So then we also perform this bioinformatics analysis and the plot where this revenue structure was formed, and this all this we have performed the study. Then we done this primer designing, cloning, and now we can see that this 930 waste pair was cloned, and then we have designing on the PCR product was forward primer, reverse primer, and this product was made, and this is then multiple sequence alignment was done, which is a cluster indicate the conserved region of clone gene XY and F1, and which is the other part is that the gene box in showing the predicted this glutamate catalytic residue glubu, and the 5 glutamine GNN residue are boxed in red. 2 asparagine and aspartic residue are boxed in blue. So these are the we have done with the multiple cellular environment, and then we have to run this ITPG induction profile of Janine protein by SGS phase, and you can see the pre-stain protein markers are there. And then lane 2, an induced sample, you can see is very high. And then density analysis determine the concentration of this particular protein. Then we also using the FELC GE, we have used the Cephalus F1 G healthcare. We purify this particular protein further, and we can see, we can found using this step, we have performed various steps and cation and bonding. So we can maximum, you can say 1.4 purification fold was achieved. So after that the purification was done, then we have to characterize this XY and F1 or recombinant XY and F1, and we can see that this XY1 is F1 is stable in almost on all pH, which is stable 3 to 10, and you can see this also stable up to 70 degrees, which is very important in commercial way, because we have to hydrolyze the things at 45 degrees centigrade, and this is also the different factors substrate which can be used for this purpose. So here we can see the stability after 12-hour incubation at a different pH, and we can determine the residual activity after a certain period of time. Similarly, we had done with the pH also, and then after 12-hour incubation, we use the metal cofactor, which can also determine the regulation of the activity, and we can determine which metal is influencing the X1 gene. So, here we can see determine the connective parameter, we determine of this, and we can see this is a metal vector and this is a Km, VMX we determine with this thing, and complete study has been published in the protein expression prediction, which can you go through the literature. Now, after this, because we had to work with the industry, and we had to give, we are not only working on laboratory, we are trying to give it, pass out the technology to the industry. So we had to work on whether this working or not. So where we made a hypothesis, if a rice straw, which can be, we are also working with the vital fungi, and lacase is the one of the periodic enzyme which can be remove the phenolic part of a lignin. So we treated it with this thing, and then we treated lacase treatment, then gyalinase treatment, cellular treatment, in-house stain, and cocktail of this. So concept was here that we use not a chemical treatment, we use the green technology using the enzyme mediated conversion and biofuel. So which can be green technology. So after this treatment, excuse me, so we have XRD FTR analysis done. Then we also determine the delignification, saccarification, enzyme toxicity, and saccarification are using individual different enzyme cocktails. So these are the I will go in a little bit. You can see substrate, which is so-called rice straw control. You can see colza lignin was around 70%. And then treated me, they have around reduction was right, lignin was that, because the lacase treatment was not so effective, because if you go for longer duration, then we're going to work, and we have to some other alternation to do. However, we can see the halocellulose in control 55 this, and treated was this 57 was this. So you can see we have conserved the actually almost after treatment of this halocellulose, hemicellulose, we do not lost this part. Okay, this is the most important. If you lost the part, we cannot convert in the sugar. The idea is that. So we have determined this all the complex system like crude gyalinase, crude cellulase, and commercial enzyme which already we are using in industry. We can the partial purified and this we have compared with this, and we can see theoretical yield the ethanol was determined. So here we can see the sugar yield sacrificial lacase treated RS complete theoretical ethanol yield and 58 degrees centigrade for 24 hours. All experiments done in a triplicate. So we had determined so how much we can be theoretical yield be there generated in a per gram liter. So this is there, and now how much we can the experimental we can do that 0.7. So we can see around 70 to 80% we can by using this cocktails and different combination we can achieve this target. So what are the consequences of the treatment of this particular enzyme mediated treatment? Then we can see determine the structure of the substrate, which is by the XRD and FTRA. The crystalline index was enzyme treated as the against the untreated as the, and this is reduced because crystallinity is a hamper to the conversion in cellulose to the sugar. So these are this ethanol production. We converted this the converted part to the ethanol, and this comprehensive study we have published and with the bioresource technology report. So these are the nowadays big market at the present time, which is people are working around the globe, and we are working on a thermal part, where the biological breakdown, and then enzymatic part, and how to develop the downstream processing, which is a efficient process, which we can reduce the cost of the products. So apart from this, I would like to thank my PhD student who are working with this from last 5 to 6 years to attend these data, and we also generated the instrument facility by DVD Fundamentals, which is a microdigester, which is working the fermenter, which we are using the rotavator, and these are working the master student which were working with me, and so. I also thank to DBT, DST, and the University Central University of Aston to provide me the facility. And thank you, and I would like to again thank you to all for listening. And if you have any question, please feel free to call me. And also you can go to the paper which I have been representing. So you can have a detailed information about this. Thank you all. Hello, is okay?
Thank you, sir. Thank you so much, sir. We are obliged to gather knowledge from you. It's our immense pleasure to have you between us, sir.
Thank you.
Now, I would like to welcome Dr. Dhruv Kumar, Amity University of Molecular Medicine and Stem Cell Research. Dr. Dhruv Kumar completed his PhD in Cellular Molecular and Industrial Biology from University of Bologna, Italy. He has been awarded as a scholar in training from Association in Cancer Research USA 2016. Apart from all research work, Sir has always been keen to spread out knowledge to upcoming generation and deliver lecture on cancer biology, stem cell biology, molecular medicine, and bioinformatics. I would warmly invite Sir on the stage to enrich our ears with your knowledgeable content. So please. Sir, you are not audible. Hello, sir.
Yeah.
Yeah, yes, sir, you are audible now.
Okay. Okay. So I will be talking about the metabolic alterations in head and neck cancer. Let me tell you that cancer has surpassed heart disease in case of killing humans. So cancer is the other members. May I kindly request please mute your mic. Yeah, thank you. So metabolic alterations and cancer is the most common, the topmost disease for human killings. And one of the important mechanism, the phenomenon that happens with the cancer for their survival is metabolic alteration. Long back, people have studied this, but the aggressive research that people have done in last 15 to 20 years on these metabolic alterations in cancer. So I will be talking about how in head and neck cancer, in a solid tumor microenvironment, as well as cancer cells, they talk to each other and they help themselves for the survival. So as you can see here, the cancer statistics in India, in men, it is the most common cancer. In women, it's a third and fourth common cancer in India. In the world, if you see the scenario of oral cancer, it's a third and fourth most common cancer in the world. Why I'm talking today, I will be discussing about the metabolism. So it's a very important to discuss at what level you can monitor the metabolic activity in solid tumor, in any kind of solid tumor. So if you see, this is the oral region of the oral cancer. Here is in the neck region, and here in the tonsil. But the most common technique that which are available today is PET CT scan. You can go to any hospital. If it is a cancer cells there, clinicians always recommend to go through this PET CT scan to detect your cancer cells or tumor in your body. And how it works, very simple mechanism is these cancer cells, because cancer cells are glucose lover. So these cancer cells trap modified glucose that we call FDG, fluorine tagged 2-deoxyglucose. So that molecule gets trapped into the cancer cells and it further gets detected by the PET CT scanner. So this way you scan cancer cells in your body. Now, this is the technique people were using to scan cancer cells. Now people are using as a therapeutic purpose. So if you want to deliver any drug to the cancer site, because cancer cells are glucose lover, you can tag a drug molecule together with the FDG. It will be delivered to the cancer cells and cancer cells will die. So lots of therapeutic implications people have done using this approach. But my idea to present this information here to tell you that how cancer cell metabolism utilizes glucose for their survival and progression. So this is the very simple mechanism. If you see how glycolysis is being regulated inside the cells in the normal cell, you have a glucose transporter here, as you can see, GLUT1, and this GLUT1 helps glucose to bring inside the cell, and this glucose then gets converted into several intermediate products, and final it produces pyruvate that goes to mitochondria to perform mitochondrial respiration, and here you get 38 ATP total from one molecule of glucose to when it gets metabolized inside the normal cell. So this is the condition of normal cell glucose metabolism. But when this normal cell changes into the cancerous, this pyruvate then gets converted into lactate with the help of lactate dehydrogenase A. And that's a problem that happens with the cancer cells. Now, let me tell you one very interesting information. You can say that if you know a little bit about glucose metabolism, that in normal cells, one glucose molecule produces 38 ATP. 36 ATP you get through the metabolism of this pyruvate in the mitochondrial respiration. Only 2 ATP total gain you get in through this process. Now, if pyruvate is not going to the mitochondria, then cancer cells are getting only 2 ATP from one molecule of glucose. So there is a difference between normal cells and cancerous cells in the sense of energy production, ATP production. So cancer cells produce only 2 ATP from one glucose, whereas normal cells produce 38 ATP. So normal cells require less glucose molecule and cancer cells require more glucose molecule for their survival and growth. And this is the reason why when people get cancer, by the time when cancer cells grow in their body, they lose their weight because most of the glucose are getting utilized by your cancer cells. Your normal cells are getting very few glucose molecule. And this is the reason why you might have seen cancer patients. By the time when cancer cells grow, they get thinner, lean and thin, very dull. Why? Because most of the glucose, the energy source are getting utilized to produce lactate. So this is the basic concept that we all study at class 6-7 level, glucose metabolism. And using that simple concept, you can control the tumor growth, cancer growth inside the body. Simply if you restrict the glucose molecule supply to the cancer cell. There are several other pathways, metabolic pathways that cancer cells utilize, but this is the one of the most common pathway and important glucose metabolism pathway that cancer cells utilize. When you talk about solid tumors doesn't have only cancer cells. This tumor contains lots of other cell types. In addition to cancer cell, you have a cancer-associated fibroblast cells here. This is the blood vessels through which your cancer cells get glucose or other nutrient. You have immune cells, you have macrophages, several other cell types that you have in solid tumor. So another failure for the cancer treatment is if clinicians, they give medicine for the solid tumor, and if that medicine is targeting only cancer cells, not other cell types which are present in the solid tumor, then you will be able to reduce the tumor size only 20, 30, 40% maximum, not more than that. So now people are using different therapeutic approach that we call combinatorial therapeutic approach. I will be discussing those things in the later slides. How you can target, these things are called tumor microenvironment, cancer-associated fibroblast, cancer-associated macrophages, all these other modulated immune cells. If you dissect the oral tumor, solid tumor, if you do the histopathology, you will be able to see the structure of that tissues like that. These are the areas of cancer cells, and these are the areas of non-cancerous cells. But non-cancerous cells are also present in the solid tumor. And these cells are tumor-associated fibroblast cells, or you can say cancer-associated fibroblast cells, that is tumor microenvironment. In case of oral cancer, breast cancer, pancreatic cancer, skin cancer, these tumor-associated fibroblast cells or cancer-associated fibroblast cells are more than 50%, 50, 60, 70, 80%. In some of the tissues, you will get up to 80% these cells. So the big question is that what these cells, the cancer-associated fibroblast or tumor-associated fibroblast cells are doing in the solid tumor. Are they helping cancer cells to grow and proliferate, or what they are doing? So we have done some investigation to find out actually what it does in solid tumor. So this is the schematic representation. If you can see this model, there is a hepatocyte growth factor. This factor is a small protein gets secreted by tumor-associated fibroblast cells, which are present in the solid tumor of head and neck cancer. This is a ligand for protein which are highly expressed, that is receptor tyrosine kinase, that we call c-MET. And this is a ligand for this protein. And when this ligand, hepatocyte growth factor, secreted by this particular tumor-associated fibroblast cells, it comes in a tumor microenvironment, and the protein which are overexpressed on the cancer cells, it activates that protein. And this activation of this c-MET leads to the increased glycolysis. And that glycolysis generates 2 ATP that helps cancer cells in a cell proliferation and progression. At the same time, these cancer cells also secrete another growth factor in tumor microenvironment that is called basic fibroblast growth factor. And very interesting, this basic fibroblast growth factor is a ligand, is a key for this lock that the protein that you have expressed on the FGFR, fibroblast growth factor receptor on TAF cells. So what is happening here? As you can see, this ligand means this key comes to this lock and it opens this lock, and allows all these metabolic pathways to get regulated. And then this metabolically active cells, this produces this basic fibroblast growth factor, which is a key for this law, and it activates. So this cycle goes on and on and on. And when these cycles are on, tumor growth happens and head and neck tumors progression happens. At the same time, when cancer cells, they do more glycolysis, they produce lactic acid. And lactic acid, through this monocarboxylate transporter 1, it comes into the tumor microenvironment. An interesting thing is that this lactic acid has a low pH. Theoretically, if tumors are having low pH, then tumors should get degraded and cancer cells should die. But what happens in cancer cells? It's a vice versa, opposite. Because even the production of, after production of high lactic acid, cancer cells grow and proliferate. What happens to this lactic acid? This lactic acid again gets utilized by these cancer-associated fibroblast cells or tumor-associated fibroblast cells, and lactic acid gets converted into pyruvate to produce more energy in these cells through oxidative phosphorylation. So we have done initially some bioinformatics analysis using TCGA database. As you can see there, when we analyzed TCGA database, we found that the c-Met expression in these cancer cells versus normal cells, the c-Met expression is very high. This is HPV positive, this is HPV negative cancer cells. Let me tell you that human papilloma virus is also associated with head and neck cancer, specifically with the oropharyngeal cancer. Okay. And when we monitor, when we saw this c-Met expression in cancer cells, we saw a lot expression. It means this gene is doing something. And we further analyzed the expression of metabolic related genes in those TCGA database, and we found that when c-Met expression is up, other metabolic related genes, hexokinase 2, LDHA, SLC2A, SL16A1, these all are upregulated. It means c-Met is directly correlated with these metabolic related genes. So as you can see here in this heat map, specifically with the LDHA, phosphofructokinase 1, MCT1, GLUT1 expression is very high. Okay, so it means these genes are doing something with these cancer cells metabolism. So we did Western blot analysis of this hexokinase 2. It converts glucose to glucose 6 phosphate. As you can see here, the hexokinase expression gets inhibited with the specific inhibitor of hexokinase here. And in the cancer cells, we saw a high expression of this hexokinase 2. Next, we did co-culture of cancer cells with cancer-associated fibroblast cells. When we put these two cells together, as you can see here, the c-Met expression, this MCT1 expression that we call monocarboxylate transporter 1. The monocarboxylate transporter 1 in the co-culture is high, okay? And when we inhibited this c-Met, we saw the MCT1 expression is low. Further, we investigated the basic fibroblast growth factor secretion through the cancer cells. As you can see here, with the condition media that we collected from tumor-associated fibroblast cells, it induces basic fibroblast cell growth factor secretion, as you can see here as well. So there is a direct correlation with c-Met, glycolysis, and basic fibroblast growth factor secretion. In the next slide, you will be able to see how this hepatocyte growth factor comes from the tumor-associated fibroblast cells, and it activates the c-Met, which is present on the cancer cells, overexpressed on the cancer cells, and it further activates the glycolysis through the activation of hexokinase 2 and produces the basic fibroblast growth factor secretion. Further, we have investigated the expression of MAP kinase. It plays important role in oxidative phosphorylation in cancer-associated fibroblast cells. As you can see here, this is the MAP kinase expression in the cancer-associated fibroblast cells treated with the conditioned media that we have isolated, collected from the head and neck squamous cell carcinoma culture. And we have used a specific inhibitor against this FGFR that we call fibroblast growth factor receptor. So after inhibiting this, as you can see, MAP kinase expression is going down when we inhibited this FGFR. So FGFR is controlling MAP kinase expression. Okay. And so as you can see here, when this FGFR, BFGF is getting secreted, it activates the FGFR on the cancer-associated fibroblast cells, and it further activates MAP kinase. MAP kinase and FGFR is directly linked. It controls, FGFR controls the MAP kinase expression. Also, this FGFR controls the oxidative phosphorylation, mitochondrial respiration in tumor-associated fibroblast cells. And this expression leads to the secretion of hepatocyte growth factor from tumor-associated fibroblast cells. So what happens together? This hepatocyte growth factor helps in the activation of c-Met here. Basic fibroblast growth factor from cancer cells activates this FGFR here, and together they help in tumor progression. So what we did, we used a specific inhibitor against FGFR, that is AZT4547, and a specific inhibitor for c-Met, PF 202341066. This one is now FDA approved for lung cancer. It's being given crizotinib, and the name of crizotinib, this medicine is available. So as you can see here, when we inhibited both receptors, FGFR in tumor-associated fibroblast cells, this is a mouse model that we used, xenograft mouse model, and we treated mouse with using combinatorial therapeutic approach. So alone FGFR inhibition doesn't, it did not reduce a lot this tumor burden, but if you can see with the c-Met expression, this is the c-Met inhibitor. So when we use this combined drug together on the mice model, we saw a lot of reduction in the tumor size as compared to the control. It shows that in solid tumor, if you have this SGF and it activates the c-Met, and also it promotes the lactic acid production, basic fibroblast growth factor secretion, and further this basic fibroblast growth factor secretion activates FGFR, and this activates MAP kinase, and further this cycle goes on and on and on, and it helps in tumor growth and tumor progression in head and neck cancer. So therapeutic point of view, you can inhibit this lock. This is the entry point. This is the entry gate for this whole metabolic activation. And this is the entry point for this MAP kinase oxidative phosphorylation activation in these cells. So you put a checker, you block these two gates. One is here, another one is here. Then you will be able to block this whole tumor progression. And this is what we have done. We have used this inhibitor, a specific inhibitor, crizotinib, against the c-Met, and then we saw the inhibition of all this hexokinase, glycolysis, lactic acid production, MCT expression, basic fibroblast growth factor expression in cancer cells. And at the same time, when you block this FGFR protein here in tumor-associated fibroblast cells, you will be able to block this process, this signaling, MAP kinase signaling, oxidative phosphorylation, SGF production. So what you are doing, you are not using the treatment approach, just a single treatment approach to inhibit the cancer cell growth, but you are using double treatment approach that we call combinatorial therapeutic approach. You are using two inhibitors, means you are targeting two receptor proteins in the same solid tumor, okay? And then you are inhibiting the tumor growth and tumor progression through this approach. So this is a very, very innovative approach that we have applied, and we have published this work in a cancer research. And in conclusion, head and neck cancer cells are highly glycolytic, and tumor-associated fibroblast cells helps in the cancer progression in head and neck cancer. So how tumor-associated fibroblast cells helps them? Through SGF and c-MET axis. And then in cancer-associated fibroblast cells, BFGF and FGFR axis helps in the mitochondrial respiration. So when you use this combinatorial approach, you will be able to reduce the tumor size or tumor growth inside the mice model. Up to that level, we have worked on it. So these are the people that work in our lab. We have 8 PhD students. Those are working on a different areas of cancer, including breast cancer, GBM, glioblastoma, oral cancer. And these are the trainees, that those who come for the training at Amit University in our lab. They have also contributed in this work. And I would like to acknowledge our collaborators. So we get patient samples from Lady Harding Medical College, and other collaborators are here, those who provide cell lines and other supports in our research. And also would like to acknowledge the funding agencies. We have right now SCRV funded project on cancer cell metabolism. Then we have received funding from ICMR, BRICS, and also the previous funding support we had from NIH USA. So with this, I would like to stop here, and thank you very much. I would like to quote a statement given by Dr. David Sherburn. He has written a book that is called Anti-Cancer, and that is totally based on cancer cell metabolism and how your dietary factors can help you to reduce the cancer burden in your body. If you limit the glucose supply, then half of the things are done. Means half of the cancer cells will die automatically. Only those cells will survive, those who are robust. For that, you take the medicine. But what happens in present scenario? We do not control our diet, because cancer cells are glucose lover. If you control glucose, you will be able to control the tumor growth and progression. So with this, I stop here, and thank you very much. And I would be happy to take any questions if you have.
Thank you so much, sir, to enrich me and the audience with such enthusiastic words. So we have a question from audience.
Sure.
So why are neck and head cancer cells more glycolytic?
Yeah, because not only head and neck cancer cells, but almost all the solid tumors, the cancer cells in the solid tumor are highly glycolytic. The one reason is their mitochondrial functions are, the impaired, means it's their genes are mutated or something. Their mitochondria do not work well, and that's why cancer cells utilize the other glycolytic pathway, that is lactate pathway, to produce energy. Okay, and let me tell you another thing. glycolysis, the rate of glycolysis is 100 times faster than the mitochondrial respiration. So cancer cells utilize this fast mechanism to produce energy as compared to the mitochondrial respiration. And that's why they grow like a bubble. These are not hard cells. They are very soft cells. When you touch tumor, it's a fluffy, it's not very rigid. It means they means whatever the things, they grow faster, they are not that robust. And this is the reason, because the impaired mitochondrial functions, cancer cells utilize glycolytic pathways to produce energy.
Thank you, sir.
Thank you.
So there is one more question.
Yeah, sure.
Sir, I would like to ask, what is the feasibility of this whole thing, the glycolysis process that is happening? We have done it experimentally, but when it comes to the hospital, what is the approach, and how much it is feasible, and what are its effect there?
Yeah, so actually there are, as I mentioned earlier, crizotinib, that is the inhibitor for c-Myc, is already in the hospital. It's already in the clinic. So clinicians are prescribing that medicine to the patients. There are several other inhibitors that blocks metabolic pathways. Metabolic pathways means, in a very simple language, means the energy source through which cancer cells, they get food. So if you block that sources, cancer cells automatically die. So what can be done? What are the targets? What are the variables that you can identify in those pathways? One is the receptor proteins that helps, that is the called GLUT protein. That's a transporter protein that helps glucose to bring inside for in the cytoplasm. If you block, there are GLUT inhibitors, you block that, you will be able to stop the glucose transportation inside the cell. Second, once glucose enters inside the cell, which enzyme helps them to get metabolized? It's a hexokinase 2. So there are inhibitors, there are medicine, there are drugs which are clinically approved drug. One can give that medicine, but at the same time, you have to be very, very careful. Normal cells also utilize the same enzyme to metabolize your glucose. So you have to be very, very selective. But when you come to the LDHA, what it does, it converts pyruvate to lactate. And that enzyme is highly expressed in the cancer cells only. In normal cells, you have very less expression of that enzyme. So what you can do, you can inhibit those enzymes which are exclusively overexpressed in cancer cells only, not in the normal cells. So those targets can be used, inhibitors can be designed against that to develop a therapeutic molecules. And there are certain proteins that we have already identified, as I have already mentioned in my talk, and people are already getting those inhibitors as a therapeutics molecule to cure the cancer through inhibiting metabolic pathways.
Thank you, sir. We have one more volunteer with the questions.
Yes.
So my question is, how daily life affects cancer, or how cancer is.
Yes, please.
So how cancer.
So your question is how your lifestyle affects
Yes,
the cancer growth
affects
and
cancer.
yeah, this is a very, very important question for all of us. And why I say it's a very, very important, because we all have cancer cells in our body, but it appears only in those people, those who have disturbed lifestyle. What is the meaning of disturbed lifestyle? What does it mean? It means that we are close to the nature. Okay, humans, if you see whatever the everyday activity that you do, if you see that that activity is going away from the nature, means you are disturbing your lifestyle. So the first thing is our everyday activity, means our in the morning when you wake up, you have a certain hormone levels up and down regulated. Are you able to get the good sleep to balance that hormones? No. If you are having some kind of sleep disorders, then your hormones will get dysregulated. If you are stressed, your stress hormone will get, overexpressed. If you are taking all the chemicals through your diet, your enzymes in your body will get dysregulated. If you are getting mutants through your drink, through the air, all these carcinogens, okay, pollutants, that may cause mutation in your normal cells. Radiation, lots of time I see people get exposed to the mobile phone, laptops, and other instruments, or the tools that radiates huge amount of radiation. That also disturbs your normal rhythm of your lifestyle. So there are hundreds of things that affects your lifestyle, starting from your dietary supplements to the water that you drink, air that you breathe, the tools that you use in your everyday life, including the medicine that you take. There are several medicines people they take for some disease. The side effect of that medicine induces mutation in the normal cells, which leads to the cancer development. Mobile phone induces brain cancer. So pesticides that you drink, that you take through the cold drinks, Coca-Cola, whatever, the Pepsis and whatever soft drink that you drink, it has a huge amount of pesticides. But that's a matter of awareness. If you are aware that you should live close to the nature, you are not going to utilize most of the things. And those things can be avoidable. And you will be able to reduce the cancer growth and cancer burden. So this is a matter of awareness, how one is aware about the things which are, surrounded to you in your house, in your office, in your college. Okay. So yes, lifestyle is one of the important factors that induce, the cancer. Apart from that, there is a genetic mutation. You might have heard about the Angelina Jolie. Angelina Jolie got his complete breast removal. She had a BRCA1, BRCA2 gene mutation, and that gene mutation she got from her mother and maternal aunt. So when she had this next generation sequencing, she found that her BRCA1 and BRCA2 is mutated. And immediately she asked her doctor to remove the breast so that she will not be getting breast cancer in future. So there are several things associated with the cancer growth and progression.
Thank you, sir. So, one more question from another volunteer.
Yeah.
Good afternoon, sir. So my question is that what is the difference between, sir, head cancer and brain tumor?
Okay. So head and neck cancer and brain tumor is totally different. Head and neck region comes from your lip. Okay. So it starts from your lip, it goes through the nostril, the nasal chamber, then it comes to the pharynx and larynx. Okay. But brain cancer it appears in the brain region. So head and neck, head doesn't mean it means the brain. Okay. Brain cancer is totally different. That the region which are associated with the brain, and head and neck region is associated with starts from the lip to the tonsil, nasal chamber, oral cavity that you have mouth. The oral cancer also comes in the head and neck cancer. Okay, so that's on the basis of the site we define the cancer types. Because the origin of head and neck cancer is the head region, because the whole region is called head. Here, from here, it's not this one is not a brain. Your brain is here, but the brain cancer is different from the head and neck region. So the definition that we define, that we give based on the origin, based on the site of the cancer.
Thank you, sir. It was very helpful. Thank you.
Thank you.
Thank you so much, sir. It was really very informative session. Moving forward,
Thank you.
I would now like to invite Professor Dr. Sushil Jha, School of Life Science, JNU, to the platform. Professor Sushil has been a research associate at University of Pennsylvania, USA. Sir is specialized into neuroscience and has recently awarded as Scopus Young Scientist. I would like to warmly welcome Professor Sushil Jha on the platform. Thank you, sir.
Okay, thank you so much for inviting me for such a wonderful conference. Let me share my screen. Is my screen visible to everyone?
Yes, sir. Yes, sir. It's visible.
Thank you so much to the organizer and especially Professor Tripathi, who has invited me to deliver my talk. That is, I'm going to talk about why do we sleep? What sleep does to our day-to-day life. I was carefully and very patiently, I was listening what Professor Kumar was saying about the lifestyle and the relation with cancer, how lifestyle changes, and he mentioned about sleep. I didn't know that he's going to talk about a little bit about sleep and lifestyle. Now I will take on from him and I'll try to emphasize how important this sleep is. So, if you ask me, can we define sleep? It's a very tough task because sleep, generally what we see, people sleep during nighttime, human I'm talking about, and several other animals as well. So we go on bed, close our eyes, and if someone is sleeping, but we can see, oh, okay, person is on bed, eyes are closed, not moving, we say person is sleeping. But if person is really sleeping or not, looking at the posture, looking at closing eyes is very difficult. So definition-wise, it is difficult to define sleep. But what happens during sleep, if we record brain waves, we can precisely know if the person is sleeping, and even if the person is sleeping, in what state he is. Because sleep is generally classified in human like in 5 stages. That is S1, S2, S3, S4, and S5. But and that is broadly if we take, we classify them into 2 stages. One is called non-REM sleep, and other is REM sleep. What REM and non-REM stands for? REM stands for rapid eye movement. So it's a very unique stage of sleep, and it has been discovered in 1953, and that was found that person is sleeping, but during that sleep, eyes are moving a lot. And also what happens when the short form is REM, rapid eye movement, and in another sleep where eye movements are not much, so that is called non-rapid eye movement sleep, means eyes are not moving a lot. Another paradox associated is during REM sleep and non-REM sleep, muscle, like what you see here, here in this case, this is a wake condition. EEG is low voltage, high frequency waves, a lot of muscle activity. Anyone can recognize this is a wake state. But when the person goes from wake to non-REM sleep, EEG becomes high voltage and low frequency waves are much more prevalent, and muscle activity is down, and that one can obviously feel that, oh, we are sleeping, muscle activity is down. But what happens during REM sleep is EEG becomes almost very similar to wake condition, low voltage and high frequency waves, but the muscle becomes atonic, means you if you are sleeping, and if you ask me how we can say by watching, but if you can say like you are traveling and sitting on a bus or train and out of a flight, and you are sleeping in a sitting posture, and you will see that, okay, one point of time your head is bending down and there's no control and you wake up. So that is the state when as muscle becomes atonic, postural muscle, we do not have a control on our head. So muscle atonia is a characteristic feature of REM sleep that happens only during REM sleep. Why all muscles except the breathing muscle is atonic, means there's no muscle activity. It is almost close to paralytic condition. Why we have that sleep state, and why we go in that kind of sleep state, we do not know. But overall, if you talk about non-REM and REM sleep, 80% time we spend in non-REM sleep and 20% REM sleep. And most of the dream that we experience every night, that comes during REM sleep. That is another interesting feature. So this was about sleep and brain waves. If you record, you can very precisely, you can determine at what time a subject was at which sleep state, whether it's non-REM sleep or REM sleep. But I'm not going to explain much about sleep. My lab is interested, and we are working on to find out why nature has evolved sleep, what sleep does to our brain and body. And if you look at this slide, you name it, the function, you name it, energy conservation, yes, during sleep, our system, body, it conserves energy because we rejuvenate our system. It has a role in immune system. It enhances your immune system, boosts your immune system. It has a role in brain development. It has a role in learning and memory, that we learn every day how memory gets consolidated. This is what my primary area and my lab is working on. Synaptic plasticity, yes, we are also working on that. So restorative function, brain detoxification, you name it, any function that you name it, sleep has a role. And what Professor Kumar has pointed out, a lot of hormones and enzymes, they are getting altered if sleep is not attained properly. I would like to give this information to the general audience. Growth hormone that pituitary secretes is only released into the system, into our body when we sleep. Growth hormone is only released when we are sleeping. So when you get up after sleep, you are not the same person. Growth hormone must have brought some changes in your body. You are not the same person. And if you are not sleeping properly, not sleeping on the proper time, even then growth hormones gets altered. And not only growth hormone, there are several, what Professor Kumar has pointed out, rightly pointed out, there are changes. So as I said, that our lab is interested in looking at how sleep helps in memory, and that is my topic I propose, and why sleep is favorable condition for memory or learning. I will try to give you some data on that, and you will understand why sleep is chosen by the nature for memory consolidation. So first thing, how sleep helps in memory consolidation. So there is a two model that we are using in the lab, It is called associative learning, means one is fear conditioning and other I will give you example of appetitive conditioning. So fear conditioning in India, we have a general कहावत है कि दूध का जला छाछ भी फूंक फूंक के पीता है. The best example of fear conditioning. General example, if I give to the general audience what fear conditioning may be to the general audience. Suppose if a house, one says that this house is a haunted house. It's a secluded place, there is a house and it is a haunted house and it is all abandoned. So no one would like to go inside because with the perception that there may be a ghost. But no one has seen that ghost, but still we are fearful. We don't want to go inside. So that is the example of fear conditioning. A general, I'm just talking about. So in animal model, what we do, we keep the animal, we bring, this is the video I will run. We bring the mice in this chamber, and it's a greed chamber, and we acclimatize this before training. So animal feels familiar with this environment. And on the training day, what we do, we give mild electrical shock for a few, like 2 or 3 times for less than a second, mild electrical shock, and then we remove the animal. And the next day, we bring the animal in the same chamber with the same bedding. What you see, the bedding and cotton, what you see, this is the reminders are there. We don't give shock. Animal shows fearful response. And fear, what we quantify, that is freezing. Means if we are scared, body freezes. So what you can see in this video, this animal is moving, the fast forward video. And you will see this animal will jump 2-3 times when the shock appears. This is the normal movement, and you will see jumping sometimes when the shock will be given. Yes, that shock was given that way, and like 2 or 3 times we give. And next day, okay, I should run this. And next day, what you see, this animal was brought in the same cage, and what you saw, the another animal is so active, and this one, a testing day, animal is not active, it's not showing, it's so fearful. And this camera captured it, and software does this kind of analysis of how much time animal showed freezing response. Animal did not move for more than 2 minutes. So that we quantified. And after this, we divided the animal into 3 different groups. One was allowed to go into the animal colony, and they were allowed to sleep properly, whatever the general lifestyle they had. So that is we call non-sleep deprived. Another animal, what we did soon after training, we did not allow this animal to sleep for 6 hours only. After training, 6 hours only, we did not allow them to sleep. Means there was a proper handling, gender handling. Then we put it on the rotating wheel, so the animal was moving on that. And another group, there was a like. I'm sorry, is somebody saying something? Am I audible, or someone wants to say something? Hello?
Hello.
Yeah, is it okay, or I'm hearing some sound somewhere?
Hello, sir. You are audible, sir.
I'm sorry, I'm audible? Am I audible?
Yes, sir. You are.
Okay, should I go ahead?
Yes, sir.
Okay, thank you. So The another is the stress control. 6 hours we keep the animal in a very constrained area, very restricted area. The animal has like a little bit of movement, so that stress we gave. But that stress, the kind of stress we chose, was not like a sleep deprivation kind of thing. So they slept, and we monitored their sleep as well. So what you see, this freezing response in a non-sleep deprived and stress controlled animal is during testing day, they showed that freezing response, but sleep deprived animal did not learn that environment was scary. So that causes memory deficiency or deficit or impairment in this sleep deprived animal. And this is what you can see, the freezing response. And then these normally trained animals, when we monitored their sleep, means wakefulness, non-REM sleep, and REM sleep after training and testing, this is what you can see, training and testing. So this is the hour change in the wakefulness amount, no change in wakefulness amount. But interestingly, what we found that the trained animal on the training and testing day showed a higher amount of non-REM sleep, that too only in a specific window after the training and testing, that is around 2nd hour to the 4th hour. That time, the amount of non-REM sleep was higher, but surprisingly, REM sleep, what I talked about a little bit surprisingly, REM sleep was significantly down in the on the training and testing day in this fear conditioned animal. Another model, this is like there is a shock, means fearful memory. Now it is a positive in reinforcer. Like what we do here, we offer fruit juice, but animal will be given fruit juice at a specific time, and animal has to learn that timing when the fruit juice will appear. So animal is inside the cage and the juice is outside, and there's a lever here, what you can see at this tray, there's a lever that brings the fruit juice from this tray up into this window. And animal has to learn what time this juice lever will come up. And the timing is, the predictors are that there is like a house light, what you see here. I will run this video. This house light will be on, then it will be off, and then again it will be on after a few seconds. So what you can see, I started this video. Sorry, I will do it again. So this house light is on, and that time the lever is not on. You can see, you can focus over here. Then light is off, and animal is alert. And see, this light is on and waiting for the juice. And when the juice is coming over there, so this poking, every time animal is poking, computer registered this number of entry. How many times animal has poked inside for the juice. Now the juice lever is down, light is also down. So this prediction is like, it is a training, it is a false prediction, but no juice. And what you can see, this entire area, animal has learned that this area, the juice will be appeared. So animal doesn't go, is always alert. You can see the alertness. So this is how animal learns. And after this kind of training, you can see the animal is moving, but when they will not go away, I'm waiting for their turn, the lever to see. So this kind of training when we give, and after that we saw again sleep deprivation, non-sleep deprived group, and also stress control group. So what you see here on the testing day, the non-sleep deprived and stress control group, number of poking was even more during testing. But sleep deprived group, they did not learn this prediction time, what time the fruit juice will appear. They had a problem, sleep deprived. And sleep deprivation is only for 6 hours, that too mild gentle handling and through wheel rotation. Another group, after training, what we recorded sleep parameters. Here, what we found, no change in even non-REM sleep. Even hourly, we did not found any change. We did not find, but what we found, change in REM sleep. So on the training and testing day, what you see, the REM sleep amount is significantly higher. So what it says, in one form of learning, non-REM was up, another form of learning, REM sleep was up. But in both the condition, it appears that sleep is more when the task was learned properly. And this happens with human as well. Now the question is, if sleep has a role in memory consolidation, what sleep does, why sleep was chosen as a favorable condition for memory consolidation? Because what we learn not during sleep, we learn during when we are alert. But the memory gets consolidated when we attain proper amount of sleep. So to find out, we have these four areas: candidate genes, epigenetic regulation, and then neurogenesis. You know, it's like in number of neurons in our brain is fixed. When baby is born, up to 4 or 5 years, neurons are generating, new neurons are formed. But after that, division in neurons are stopped. It is not like a cell division in other body, other organ, like liver. Liver keep on generating new cells always. Muscles, new, always. But brain has a limitation. The number of neurons is fixed after 4 or 5 years of the birth, and that remain fixed lifelong. And with time, the number of neurons are going down. Some neurons are dying. But in some part of the area, the neurogenesis is taking place, and that is called adult neurogenesis, and that happens in two areas of the brain. One is in the hippocampal DG area, dentate gyrus, and another in the subventricular zone. So I will just discuss these 4 points that helps sleep to consolidate memory. So first, to focus on some of the candidate genes, we focused on these 4 genes, FMN2, GSK3 alpha, and CTN and CHAN3, because what we found that after training, especially in fear condition learning, what you see, the expression level of FM2, GSK3, and sorry. Okay, so you can see my, this arrow. So what you see, this 1 hour after the training, this level of FMN2, GSK3, and CHAN3 are significantly up. Even that is the 3rd hour also, only CHAN3, but other came to the normal level. And CTN, the level was little bit down, significantly not up. But all these 4 genes, what we found, that when we knock out, what is going on? So when we knock out with shy RNA, small interfering RNA technology, so you can see knocking out these 4 genes with shy RNA, blocking their activity, this animal did not learn. Person freezing was significantly down compared to the vehicle and the negative control. So it shows this level after learning, whatsoever the expression level is, that is very, very important. And when we train the animal and we sleep deprive them, then we found that level was significantly down. So all this level was down. So sleep deprived condition did not allow proper amount of this gene expression, or their protein level should be at that level after learning. So sleep deprivation is altering with expression of gene in the particular brain area where this memory is getting consolidated. And that is the dorsal hippocampus. So we did it in the hippocampus. And gene expression, you must have learned about this epigenetic regulation. There are two enzymes, HAT and SDAC. So acetyl transferase, histone acetyl transferase. When acetyl group are attached, the linker portion of the DNA will be more spaced out, genes are expressed. And when the acetyl groups are removed by the enzyme histone deacetylase, they are now again condensed and the gene expression will be blocked. So the level of this HAT and histone deacetylase is playing important role in memory as well, memory consolidation. So what we did, the kind of approach that we took was we blocked the activity of HDAC. So this Verinoset is a drug that blocks the activity of this histone deacetylase. And when we block this activity, what we found that after fear conditioning, this is what the acetylated H4K12 level is. So the baseline, and then after training, this has gone up. Even after training in a stress control animal, the level was high. And that predicts that why these two group animals did not show learning deficit. But in sleep deprived group, what you see, the expression level is significantly down. But when we injected SAHA in normal animal, now you can see this acetylated H4K12 level. And in sleep deprived animal, after training, when the SAHA was injected, the level remains high. And the SAHA injected animal did not show any learning deficit. So we can say that if someone is sleep deprived, and during that time, if SAHA is present, they will not be facing any memory deficit. That could be, but then it is a drug. And Professor Kumar has rightly pointed out, drug is not going to favor us. Long run, there will be a problem. Coming to the third question, if sleep has any role in potentiating the neurogenesis rate, yes, answer is sleep helps in potentiating the rate of adult neurogenesis. So what you can see, this small dots here, this one, this one, this one, these are all BRDU leveled cells. BRDU is the analog of DNA, uracil, and then it gets incorporated into DNA. So when cells are dividing, these will show the signal fluorescence. So under microscope, you can detect. So all these are newly formed cells because their DNA is marked with BRDU. And now you see in this animal, the number of cells, BRDU positive cells, you can compare. So then after training, when animal gets proper amount of sleep, or we do not alter there. So after training, the newly formed neurons or newly formed cells are significantly high. But when we sleep deprive them, now you can see it is back to the basal level. So the training induced immune cells are now hampered by sleep deprivation. So you can see how this is the DCS positive cells, means all these newly generated cells are going towards either, like they have a fate, they can go to form glia or neuron. So DCX is a marker of newly formed neurons. So now you can see how these newly formed neurons are incorporated into the system after training. So if you ask me what happens to human, what we learn, obviously it increases, accelerates the rate of neurogenesis. So neurogenesis is another mechanism. After training, it goes up, and that helps us in memory, to keep memory for a longer period of time, because these are the neurons, they are generated in response to learning. So they are going to keep all the information because they are generated in response, getting matured in response to training. So information is going to stay over there for a long period of time. But if sleep deprived condition, back to the normal, to the basal level. So obviously learning will be impaired. And these are the certain markers, certain factors that also helps in potentiating neurogenesis. For example, this level of phosphorylated ERK1 and ERK2. Now you can see in dorsal and ventral hippocampus after training, two different tasks, race conditioning and delay conditioning. Their expression level is high, even Krebs level, phosphorylated Krebs level. Not like a general level did not change, ERK1, ERK2 and Krebs, but phosphorylated level has gone up. R is a marker of neuronal activity. So their level increases means that shows the activity of the neurons after training. So neuronal activity increases, and it has been found that these activities are replayed during sleep. I will show you that data. So all these suggest that genes are expressed after learning during sleep. Then even epigenetic level, they helps in learning, and sleep helps in epigenetic regulation. Neurogenesis is up. Now the question is, this was proposed by Dickelman group in Germany, that when we learn, and if it is a hippocampal dependent task, the neurons in the hippocampus will be activated during acquisition of information. Once it is acquiring the information, that is not sufficient for the neurons to keep that information for a longer period of time, unless and until it is reactivated. So he has proposed this neuronal reactivation reinforcement theory. So what he proposed, that when we learn this information, neurons are activated, then this repeated reactivation is taking place, and it happens at several days. I'm not sure what REM sleep means, but as a general, at this moment, I'm giving a general example, then general perception. When you are preparing for your exam, we learn several things. We try to mug up several things so that we can write properly in the exam. So every words, language, their framing, everything we learn in a proper manner. And if you remember what happens at night, when you sleep after learning, you spend more time in dreaming. And when you get up in the morning, you don't feel fresh. You say like, oh my God, I was dreaming, I'm exhausted. So REM sleep occurs more frequently after, during the examination period. And during sleep, with several experiments, it has been shown that neurons are reactivated. And once you learn that replay of neural firing sequence in rat hippocampus during sleep following a spatial experience, this has been shown by Megnow group. And also what we found, this is our group data, all these lines that you see, these are neuronal activity. And this is the example of a remodeling neuron, means the visual cortex. And the example is ocular dominance plasticity. So what happens during the birth, there are neurons in the visual cortex in our brain. Those are associated with either right eye or left eye. So if you close one eye for few days, the neurons, those were associated with the earlier closed eye. Now after closing, those neurons will be shifting towards the open eye. And this is called ocular dominance plasticity. And it happens only during early stage, not after that. Means, for example, if I say, if the baby is born with cataract in one eye, so there will be impairment in their or impediment in their retina to receive proper amount of light. So that stimulus cannot go to the brain, and the cataract eye will lose connection, and all the connection of that eye will be shifted towards the good eye. So this is what the example is. We induce that remodeling neuron. So what you see here, every neuron is here, baseline condition as a post-MD condition. So it was associated with one type of the neuron and it shifted towards the another neuron. And that happens only during non-REM and REM sleep activity. You can see how it is changing. Wakefulness, the number of lines is almost comparable. So the shifting of from good eyes, from bad eyes towards the good eyes, that took place, and it happens only during non-REM and REM sleep. So REM and non-REM sleep is also favoring these neurons to judge or allow them to judge the condition, and because bad eyes, no one wants to get associated with that. So that will be shifted towards the good eye, and that is left lifelong is going to be remain like that. So that is how baby's cataract needs to be operated immediately, otherwise baby will be blind with that eye. That has been shown. So now the question is, if the neurons are reactivated during accusation and the similar pattern that follows during sleep and several times, several cycles of the sleep, not only the first day, the second day, third day, fourth day, like that. So when we learn some information, there is stimulus, neurons can be activated. But if the same neuron is reactivated during sleep, where is the stimulus? From where the stimulus is coming? So what sleep does, sleep provides that stimulus because there are certain waves in the brain, and these are called cortical slow wave activity, sigma waves, theta waves, and these are appearing at a particular time point. For example, this is the example of theta waves, sigma waves, and it is happening only during non-REM sleep. Theta waves is coming only during REM sleep. And these are all electrical waves, and it has a potential to increase the neuronal activity. So the theta peak is at, you can see this is the highest during the training and also during testing. So what you see, basal level is 100%. So these peaks are very high in the trained animals. All these are trained animals. And it happens at the cortical level as well as it happens at the amygdala level, at the hippocampal level. So if these peaks are coming at a particular time during sleep, they are going to specifically activate those neurons and provides the environment for replay. And those neurons are replayed. If it is also associated with the dream, I do not have a data, no paper has come up yet, so I cannot say, but I will not be surprised if tomorrow it appears along with dreaming. So sleep is very, very favorable condition for information to get processed so that learned information can be consolidated for a longer period of time. So to conclude, sleep plays very, very important role in memory consolidation and mechanism we talked about the way we see. Now what happens if you ask me if we sleep during daytime and we sleep, remain awake during nighttime, so proper amount of sleep we are getting, will it affect memory or not? Answer is yes. So what Professor Kumar said, we follow our cycle, the way we are born or on this planet. So if we are not following the nature's rule, we cannot sustain a proper cycle in our body. So thank you so much, such a wonderful audience. And these are my teammates in the lab. With that, I acknowledge our funding, that is CSR, DBT, DST, all those funding agency. And once again, thank you so much. If you have any question, I will be happy to answer that.
Thank you so much, sir, for such an informative session. I thank all our honorable speakers who have been with us throughout the first day of 4th National Conference on Cellular and Molecular Biology. We would continue with the same enthusiasm on the second day of conference. I request all my faculties and students to join again tomorrow for the second day of conference. Thanking you all. Please stay safe.
Okay, thank you so much and wish you all the best.
Thank you, sir. Thank you for the session.